Pero de esta de cristal de telescópica.
Afirmativo. Si lo dejas estás hablando, pero puedes bloquearlo. Entonces ya así ya puedes hablar durante todo el rato. Para desbloquearlo, pulsarlo y otra vez.
Los auriculares un día complicado. Yo esto lo único que... No, no, tremendo. Yo cuando me he hecho... Good morning, everybody. Welcome to this third edition of Microbiome. I'm very happy to be here with you today in this third edition. Microbiome is the most important event internationally and worldwide regarding agricultural microorganism, its composition. We only know 2%, and the advantages that so far are a lot of them, even though we have very little knowledge. So here today we have gathered together to take a step forward towards the future where microorganisms will be the most important players in agriculture. We welcome Carlos Garcia, who's going to open this event, officially open this event.
Good morning. From the organization of Microbiome, we have decided to open the conference in the shortest way possible because we are aware of the fact that what you are interested on, to all of you here in the room, we are around 1,000 people on site, and those online, that's like 600 people, you are interested in speakers and in the different papers. So I want to keep it short when opening this event. But as Alessia was mentioning, this is the third edition of Microbiome. And I want to start thanking some of the people who have made this possible. This is an adventure, and this is an adventure. This is a challenge because within this symbiosis between the academia and private companies, this is a challenge. In 2019, the first conference was started, and there were two people, Pedro Palazón and Pedro Fernández, who are in the room here today, and Arturo Soler from Agromarketing. They were the ones who convinced me to be part of this feat because I wasn't aware of how important it was going to be. Alessia was mentioning, and I thank you for the introduction, this was a highly interesting topic at all levels, but I was kind of skeptical. This is the third edition. The second one was in 2021 during the pandemic, and this event is the one that gathers us face to face. So despite of the fact having worked on the topic two times before, we're going to see today how nowadays we're still lacking knowledge to be able to know all the problems related to microorganisms in agriculture. And I think this is the right site for us to get to know what's going to happen in the future, especially when we face different sources of stress. We have to analyze agriculture from a scientific and technical point of view and how we can get sustainability in the future. That's why this event is going to be essential according to our point of view. Without further ado, I want to thank Pedro Palazón, Pedro Fernández, and Arturo Soler from Agromarketing and all of their teams that, as you can see, have worked really hard to organize this wonderful event to start Microbiome 2023. I've got some notes, and they have told me to give some explanations. Two people are going to be controlling, Alessia and Maria are going to be controlling times of the different speakers. So please try to stick to the time as much as possible. We know those of us working on research, as myself, we know that it's hard to stick to the 20 minutes, but if you visit the microbiome app, you can see the different speakers and you can ask questions on the applications. Questions are going to be answered later on. And you can also contact speakers here on site. And during the different breaks, coffee breaks, fruit breaks that we're going to have, we will have the time to interact. And that's the most important and useful thing of these events, that we interact. And during the presentations, please convey your ideas in the clearest way possible so that people get to know them. So please stick to the time. And the event coordinators, Alessia and Maria, are going to be really strict with the time. Something else that I've been told is that there are 2 networking rooms on the first floor. They are at your disposal for you to use them in case you need them. And during breaks, we're also going to try to be strict. And 5 times before they finish, we're going to call people to come into the room. 5 minutes. And in case there's any problem, you know that on the ground floor, you can see the technical office of agromarketing there at your full disposal. That's all I have to say. I want to thank you again. Being able to see people who visited us and attended this event in 2019 says a lot. It shows that we're doing things kind of okay. And let's hope this is useful for the future too. Without further ado, I officially open the conference. And Alessia, I give you the floor.
How can we join innovation agriculture? You have to connect with the leaders in the sector. You have to learn from experts and access new technologies. Connect, learn, experimenting. Microbiome 2023 International Conference. And now we're going to have our first speaker with us, Carlos Garcia, that is going to convey the importance of scientific research in in agriculture of microorganisms. Thank you, Alessia.
The advantage of speaking now again is that I just remember that I have to thank all of our sponsors who thought that it was useful to contribute to this event. Now, as Alessia mentioned, I'm going to talk about the importance of scientific research for 2 reasons. The first one, because I am a scientist myself, and I believe in scientific research, because without it, we're not moving towards the future. As I did in the past, and many of you know me from other events, not just from microbiome, I work on the soil science. The institute I work at, CSIC, does the Higher Council of Scientific Research at the Applied Biology Center of Segura River in Murcia. And I work in soil science. I could talk about my specific field of research. I am a biochemistry. I work on biochemistry myself. But what I want to do is to show you from my point of view where we're moving towards in the field of microorganisms and what scientific research is offering us. Again, without scientific research, we're not going to have any future in agriculture or in any other field. This is a vision of the research group that I'm in charge of, where we see scientific research from a wide scope, and I want to show you why this is essential for us. People from the academia who are here today carry out research. People from private companies carry out research, and we have to make the most of it. And this is what I want to highlight on this first presentation. Let's see if this works. So talking about the importance of scientific research after a pandemic or after the news we receive day after day that are important. For example, a few days ago, we saw a piece of news on the appearance of a drug for a very serious disease. And Moderna in the USA has programmed for 7 years a vaccine against cancer for each person. It's kind of a customized vaccine. They're going to be investing money on that line on research. So how are we going to be against scientific research? I'm sure all of us here today, we are part of a collaboration between the academia and the private companies. Then we have to talk about businesses. That's something different. About investment. That's something different. But from my point of view, highlighting research is essential. This is a dynamic process. Research may change throughout the way. This is an ethical process that generates knowledge. And what do we do on research? We describe, explain, understand all the facts that are happening in the best way possible. There are top researchers, there are other researchers that we do our job as best as we can with the resources we have. And in the scope of civil servants, that's what I work at, with a high amount of vocation. We are highly vocational. We have to research for the society to benefit from my job. And I'm fortunate to work in what I like. But there are some aspects that we have to assume. Scientific research is an experimentation, a mathematical and experimental method, but we always have to work with a defined hypothesis. First of all, we have to think what's the hypothesis we're going to be working with. If there's not a correct hypothesis and some previous premises, our research may have some gaps. Although research from the biomedical point of view may be kind of abstract, if we move to agricultural sciences, we have to take into account that agricultural sciences have to do with natural resources, with the environment, even with biomedicine, because at the moment we're using in agricultural sciences and in soil sciences, we're using omic techniques that are available in other branches of science, in other specialties or areas of science. And I'm proud of working in science to contribute with knowledge for an activity that is key for our continent, for our country and for the world. Offering something for agriculture to develop in the current world through research, that has to make us proud. We have to be proud of it, all of us today here today. We have to start from some premises. First of all, sustainable agriculture based on knowledge and research, the need to produce more with less resources. We have to use less resources. This is a huge challenge for the future, but that's what research is for. And the third premise is what I work on, that's agriculture and the soil. If you have listened to me in previous presentations, I always try to convey the same message. Regardless of what you work on, if it's biostimulant, micromolecules, fertilizers, if it's related to the soil, it's going to end up in the soil. And the soil has to introduce that in its processes. And if you work or collaborate with the EU, those of us working on the value of the soil for years, now the EU is supporting us, saying by 2030, 75% of the soils have to be sustainable. Then we need to understand what soil sustainability means, but that's something we have to work on. Secondly, the soil have to contribute to the earth biodiversity in 25%. And that has to be a key premise for those of you here today. We carry out research. We all work with all kinds of products, except for leaf products, foliar products. And if we want plants to be taller, stronger, with longer roots, that's going to have an effect on the soil. So the soil has always have to be taken into account. So taking all that into account, we create a hypothesis. We can manage those microorganisms in the soil, things coming from inside and from outside the soil, so that we can direct it towards something that is beneficial for the soil, for productivity, and for agriculture in the future. I don't know if this is working. Okay. I'm not going to tell you what we know about microorganisms because most of you have a wider knowledge than myself. We know what they are. We know what they do. What are the processes we expect from them? What's the interaction between the soil and the plant? Then there are some conditions, but we are aware of them. What are those processes? We have to keep it clear. For some things, it's useful. For others, it isn't. I'm going to give you an example so that it's even clearer for you. It's necessary, or scientific research is necessary. And there are some aspects that we have to take into account. We need to harmonize what we know. How many times we read something and we forget about the background. Because research is not just being right. It's making mistakes and telling people in the future, don't go that way because that's not correct. So for many people here at the business level, this is basic. This is what we have to take into account, what I want and why I want to get there. There are two things that are really interesting for me. That's really related to the topic of the soil. What about interactions? If I add something to the soil, don't you think that biomolecules that I'm going to put in the soil, it doesn't exist already on the floor, on the soil, sorry, how is it going to interact? Are there going to be synergies? This is a key condition to know why I did that experiment and it's perfect, but then it changes. First of all, the soil is highly complex, and I've never understood why we all think that behavior of the soil is the same if it's clay, if it's sand, if we're working with a plant or another one. So interactions have to be taken into account, and we have to accept that this is holistic. Agricultural sciences do not focus on a specific area. It works with ecology, with microbiology, within specific conditions that we can see. Therefore, in the future, we have to take everything into account. And in the end, we finish in the soil microbiome. Soil microbiome, it's extremely complex. It's not just a matter of microorganisms. They have to be specifically ordered if we want it to work the way we want it to work. And in order to improve and to keep biodiversity and never intervening in a negative way. Maybe we get plants to grow faster, but what are going to be the consequences in 3 years' time? That's something that we really have to bear in mind. In the bottom of the slide, you can see a paper that was recently published regarding soil microbiomes and climate change. Because if all of these we're going to be working with today and tomorrow, if we do not make the most of microorganisms to fight climate change, we're going to be on the wrong path. Simply, this is on our research, we really need to know processes. Research is not just a matter of this has grown more or less. What happens to processes? Let's think about phosphorus on the soil. The phosphorus is non-soluble on the soil. But you also know that the plant cannot take it. So what is it going to happen? I'm going to use matter to make it soluble. Then there are phosphatase and there are other aspects that are essential for plants to assimilate it so that the plant can use those substances. When I do that and I use a microorganism, I know that the plant is going to be able to take that substance. Especially talking about bioagriculture. So we have to know about it. We have to increase our knowledge about ecosystemic services and processes. How can plants get carbon from the soil, for example? I'm sure many of you are more familiar with agriculture itself because I work in a lab, and I'm sure we're going to see the consequences in the future. We have to keep ecosystems and soil ecosystems. We have to adapt agriculture to climate change whilst fighting climate change. There are some papers that our research group has published where we can see the importance of different structures on the soil. Importance of interaction of agricultural practices on the soil life. Do you think that when we use organic fertilizers, and I'm in love with organic fertilizers, quality organic fertilizers, because with chemical fertilizers, I'm polluting the soil. So if it's high quality, I'm in love with it. But do you think when I use organic fertilizer, it doesn't interact with all the organisms? When someone does something in an isolated way with fertilizers, of course, it affects the soil. This has to be taken into account. Because even NPK has an influence on the soil according to the different conditions. And the last point is the need to avoid risks, because the EU is telling me that the soil has to be healthy. We have to create protocols for our interaction not to affect the carbon and the nitrogen process. So we can say, yes, I'm intervening on the soil with these products, with these conditions, but I'm not negatively altering the soil, and that's going to be compulsory. If I want a microorganism, a biomolecule, to be efficient, the soil has to be as healthy as possible. These are the minimum conditions for a healthy soil. But in my presentation, there's a paper that specifies what a healthy soil is and how it has to be calculated. But of course, we need to have a good soil for things to work, and we need microorganisms to contribute. We have to limit stress. I have mentioned also biostimulation. You're going to listen about biostimulation, introducing pool microorganisms. Am I running out of time, Alessia? You have less than 1 minute. My screen has disappeared. I just want to mention that biostimulation conditions the fact that I can act on the soil microorganisms if I know what they're doing. And if they work at 30%, maybe if I activate them correctly, they can work at 70%. So the soil is going to work better. These are some assessment conditions. In agricultural sciences, I would evaluate or assess, make an assessment knowing if I work in vitro or in vivo, experimental designs that are adequate. Please do not forget about controls, not positive or negative control. We need to use it because otherwise we're lost. Sampling handbooks. You cannot sample anywhere at any period of the year at different depths, and we need to establish the conditions because at the end the effects on the soil are really important. I'm not going to talk about the omic sciences that are there in the soil science journal, there's a paper on omic sciences and omic practices, and a paper with only a specific measurement at a specific moment is not valid. It's not really useful. This is out there for you to read. This is an image. We're talking about microorganisms on the soil. We talk about pools. There are biomolecules that may be important in the future for agriculture, and there are enzymes that are synthesized by microorganisms that can be really useful with the right conditions as biostimulants, and they can improve the soil health. As this is going to be mentioned today, knowledge has to be generated according to new techniques. Metabolomics may change the future on a short term. We are getting to know exometabolome, all the different consortia where we get different metabolites that can condition what are the metabolites that have a positive or a negative effect for control and for stimulation. This is one of the lines. And finally, we have to take climate change into account. We have to avoid stress, scientific research of customized products. Let's create customized products. I was talking about a customized cancer vaccine. Can't we customize products for agriculture? Do we think that all products are useful for all types of soil? That's not the case. So let's try to use microbiome for our interest, to manage soil health, to get a higher yield, to avoid diseases on plants, and to improve quality in the future. Finally, I want all of us here today to be aware on the importance of the collaboration of all of us today in the public and in the private sector. There's a lack of funding. You know how much money was invested in the COVID vaccine? Maybe we have different points of view. Maybe we have to publish papers very frequently. Those of you in companies, that's not the case. But it is really important for us to publish useful papers. So let's work together. I'm really sure that we have to collaborate. If we are able to have open spaces where we can collaborate, this is going to be a success for all of us. This is a picture of my group. We work with European and national projects, projects with companies, regional projects, and thanks to this research group. People are essential when they are part of a group that enables us to integrate research. Thank you very much. I'm really sorry for taking too long, but the opening speech doesn't count, doesn't have to be taken into account. Thank you.
We're now going to welcome Ángela Borràs from Plymag with her talk, How to Enhance Microbiota of the Soil and Establish Microorganisms in the Field. Welcome, Ángela. Good morning, everybody. It's a great pleasure for me to be here with you. Thank you, Alexia, for your introduction. I'm Ángela Borràs, Marketing Director at Plymag, and I'm here to tell you about our proposal for microorganisms. I would like to start by thanking the agro-marketing team for their enthusiasm, their affection, and the way you have organized the third edition of Microbiome. Without any doubt, it's going to be very successful, as it was the case with the first and second edition, and I hope that future editions will also take place. If we think about the great welcoming reaction for this, we have different answers. The first one, because microorganisms are a trend. We see constantly on the newspapers and journals, we see different pieces of news about the use of microorganisms and their benefits for agriculture. And Spain is leading this change in agriculture and is also the first person devoting resources to researching and to use in the field these microorganisms. The second answer, that is more kind of tangible, is to think about this potential market we have with microorganisms and biostimulants. We know that the biostimulant market has doubled in the recent years, and if we look at figures in Spain, we can see that this trend is obviously replicated. And if we take a look at the specific segment of microorganisms used in biostimulation, this market has been multiplied by 3 in the last 5 years. Certainly, this is very interesting for private companies, for research institutions to come up with new solutions for farmers so that we can respond to these needs. Without any doubt, the demands of the administration have been very challenging, and the European Green Deal or the farm to fork strategies have been very important for us to focus on sustainability and reduction of inputs. Europe is committed to reduce the use of fertilizers and phytosanitary products in the next few years, and this is a very big challenge ahead of us, where microorganisms have room to come up with solutions to this situation that we're facing. Okay. The final response is that microorganisms work. We have lots of evidence for this, and throughout today we'll see novelties regarding how they work and their benefits. If we focus on the agriculture point of view, the problem is the role of these microorganisms, how to adapt them to our agricultural soils, and how to solve the daily problems of our technicians, advisors on the field as to how to implement microorganisms in the field. Summarizing the composition and properties of soil, we know that the soil is made up of minerals, organic matter, water, and air. And depending on the origin and the proportion we find, we'll have physical, chemical, and biological properties that will provide us with the traits, the fertility traits of the soil. We can modify to some extent these properties to achieve the maximum potential of our crops. That is our final aim. However, we know that there are some agricultural practices that have led to the impoverishment of the soil, and this here is where it's fundamental to manage in a more sustainable way, to promote existing resources, to measure very well what we have in our fields, to contribute these exact supplies that we need in our for our crops. If we focus on the rhizosphere and microorganisms that already exist, we know that most of them contribute benefits, like the decomposition of organic matter that exists, the fixation of atmospheric nitrogen. They improve the structure of the soil. There are produce biostimulants, compounds, siderophores, and polysaccharides. They limit the development of pathogens and diseases. They degrade and limit toxic substances and damaging substances, and they induce the development of the root. And diverse microbiota is very important for our crops. This is where we find the potential of rhizobacteria and the use of microbacteria in the field. They're going to provide us with plant biostimulation, reduce the chemical fertilization, reduce the environmental impact. They're compatible with every kind of agriculture, ecological, organic, conventional, and they can adapt to different environments. If we know the properties of the soil, this is going to enable us to work with different microorganisms to and get the most potential of the largest potential of our crops. However, we know how microorganisms work, how they reproduce, but we still have a long way to go to find a response for daily needs of our fields. Can I mix these products? How can I coordinate the whole program? This is very common, and we faced compatibility problems and problems with the heads. And this is where we need to provide responses to our farmers. In this sense, Primark has developed a solution called Ubuntu, a single solution that is easy to apply, has high solubility and high and maximum compatibility among species. What is Ubuntu? Is based on the concept of putting together prebiotics and probiotics. So having microorganisms at the core, we can accompany them of a prebiotic that can help us install it in the field and to get the best yield and the best benefit of the characteristics it has. To be more precise, Ubuntu is based on extract from algae and different extracts that in the right proportion is going to provide us the benefits for our application. Humic extract, microorganisms, extract of algae, and amino acids. We have chosen 4 specific species of microorganisms that you can see on the screen. Why have we chosen these species? Well, we know that they have very interesting traits, such as the solubilization of phosphorus. They enhance hormonally our plants, and they help us induce the systemic response acquired, that even through the root we can have responses in the higher part of the plant. they also help us solubilize potassium, have beneficial enzymes on the soil that are going to give us a better intake of elements that we already have in the soil. This enhances what we already have in the soil and produce more with less. Azotobacter chlorococcum fixates nitrogen very well, accelerates the production of different things and of hormones, and help us fight an endemic problem in the Mediterranean. Azospirillum brasilense is going to help us promote this development of the root and the production of antimicrobial compounds. In brief, we have chosen a microorganism consortium, thinking with the idea of increasing the solubilization of phosphorus, help solubilization of potassium with the root, using the root as the core. We need to have a strong root system that is healthy to manage to increase the absorption of everything that we already have in the soil. However, not everything is worthwhile. Not all probiotics work in the same way when we mix them with our microorganisms. And this is something that we need to bear in mind when we set up this program of applications on the field. What have we done to see and to choose the best probiotic? The first thing we did was to measure the growth potential of our consortium of microorganisms. This would be the potential of growth of the 4 microorganisms that we chose in a general environment. And here we can see how these microorganisms grow when they go together with different compositions and different proportions of probiotics. This is not working, I think. Well, but you can see if you point it towards the screen, it'll probably work. That's okay, that's okay. I think you can see it very well. There are different formulations where there is a reduction in the growth of microorganisms. So as Carlos was saying, it's very important to know what we have and what we're going to add to enhance growth, because there are probiotics, extract of algae, amino acids, and humic extracts that can reduce the bacterial growth. But we have other probiotics that help us increase. It's important to highlight that the chain of microorganisms was in a combination of 10 to the power of 9. And this was in a concentration of 10 to the power of 8. So this means that we are assessing a concentration that is 10 times smaller of microorganisms. With this solution that we find, we can increase the potential of growth of our blend of microorganisms, the original blend, applying 10 times less the concentration of these microorganisms. Here we can see in a visual way, comparing the original blend and the product that was formulation 4, the one that was better, that this bacterial growth is exceeded, and we can find the 4 morphotypes that we have identified as that are our microorganisms. There's a great compatibility among microorganisms, and the prebiotic is going to help us increase this efficiency. The most curious thing about this is that not only have we increased the capacity to reproduce of these microorganisms, we've increased their biological activity. In this case, we have once again the original blend where we have measured the fixation activity of nitrogen. This is the maximal potential with microorganisms on their own, and then we have the comparison with the different formulations that I was mentioning at the beginning. Here we see that in formulation number 4, we have increased in 28% the fixation activity of nitrogen. On the one hand, the probiotic helped us increase, 2 minutes, Angela, has helped us increase this amount of units that create colonies, and we've increased the fixation or the biological activity of the latter. Here we have a visual image where we see the microbial growth and the increase of the fixation of nitrogen. And finally, I wanted to say that of all the trials we have carried out, we've chosen this one regarding the lettuce in the sea area. Here we have a control without irrigation. We thought it was very interesting to have this trait. And then we had control with Ubuntu at 100% and another one with Ubuntu and irrigation at 50%. The other one was irrigation at 100%. The results in the total development of the plant were very significant. This is the control with irrigation and non-irrigation. The irrigation is affecting us, and then we see that with Ubuntu we managed to increase the weight of the root. And the important thing is that reducing the 50% of the irrigation, we increase the plant expression of these plants with 50% of water. And in the thesis of Ubuntu, we have too much water. We could have reduced the water regarding vis-a-vis the control. We were talking about lettuce in nursery. We have now more weight at the top, more weight in the root, more leaves in the top, and we can increase the retention of water. And this is very interesting. Sorry. So to sum up, the advantages of our product are clear. They help us improve the ability to retain water. This is at the core because the results have been amazing. It's helped us to improve the plant state, the nutrients in the leaf, and a better root development. I would like to highlight the easy application of Ubuntu. It's a solid product that has high solubility and allows us to reach concentrations of 20% in the application tank in the heads, simplifying the operation for the farmer. The moments of application can be diverse depending on the aim that we're looking for in the field, the establishment of the crop or transplant in the nursery, the beginning of the root development or root sprouting, the restructuring of poor soils that we need to reach the highest potential and yield. This is going to restructure poor soils, as I was saying, and increase effectiveness in areas where the regeneration of the soil is lagging behind. And something that's very interesting is to use this kind of product in all the phases where we have a higher demand of nutrients. We're going to help this through the microorganisms in the root. To recap, your time is over. You have to set the aim very well. We cannot talk about microorganisms in general. We need to know what we want to achieve and what microorganism or consortium of microorganisms is going to allow us to reach that aim. We have to select compatible species among themselves. We can add a microorganism that is going to be more negative than positive. We have to study the best combination of prebiotics and to choose stable formulations. This is our proposal in the field of microorganisms. We will be at our stool to be able to answer any questions. Thank you very much to my team in Plymag and the development team. Thank you very much. Thank you, Angela.
Now we're going to welcome Lionel Dupuy from Naker. This paper is going to be in English. The paper is going to be in English, so there's simultaneous interpreting available at the APP, so that you can listen to it in Spanish. Thank you, Lionel. Welcome. Muchas gracias.
Sorry, this presentation is in English. Right, so our laboratory studies the rhizosphere. So the rhizosphere is this thin layer of soil that you can see in the picture that is under the direct influence of the activity of the plant.
Que afecta directamente la actividad de la planta. Es algo que podemos ver cuando accedemos al suelo y vemos las raíces. Vemos cómo la situación cambia en el suelo como resultado de la interacción de la planta con el suelo. La rizosfera es importantísima, hay muchísima actividad microbiana, es muy importante para la nutrición de la planta, ayuda a la absorción de nutrientes y a solubilizar los nutrientes, y a través de los pelos de las raíces se transportan esos nutrientes a la planta. Y es importantísimo tener un microbioma sano en la rizosfera para evitar la colonización de enfermedades y la extensión de enfermedades. Estoy seguro de que nada de lo que acabo de decir os resulta sorprendente porque estamos en el Congreso del Microbioma, pero lo que sí nos interesa en concreto es cómo el movimiento de estos microbios permiten establecerse en la rizosfera. Porque cuando las raíces tienen que establecerse y queremos saber cómo se mantiene esa rizosfera. Porque imaginemos que queremos introducir una nueva cepa en el sistema, que queremos que se expanda por el sistema radicular. Y esto es lo que más nos interesa y es lo que vamos a ver. Vale, los que trabajamos con las raíces de las plantas y los microbios sabemos que tenemos distintos métodos, porque el problema es que si queremos acceder a las raíces tenemos que eliminar mucha tierra y esto lleva mucho tiempo, tienes que extraer mucho suelo, no sé si veis mucha tierra, no sé si veis la imagen. Y entonces no queremos romper la interacción porque si no, no vamos a ver lo que queremos ver. Se han producido muchos desarrollos técnicos. Estamos hablando de resonancia magnética. Aquí vemos una reconstrucción del sistema radicular, que es la técnica más popular. También el TAC es hacer una radiografía de la zona de las raíces. Se utilizan distintos tipos de radiación y este sistema es muy bueno para el agua. Esto es muy bueno para ver los microbios, pero claro, son muy, muy costosos. Y también exige que la gente esté formada, etc. Por eso la primera parte de mi charla no aparece. La primera parte de mi charla es un método que hemos desarrollado para ayudar a realizar el seguimiento de las interacciones de las raíces con la rizosfera. Y esto se basa en la idea de Smart Soils, que son suelos inteligentes. Son suelos artificiales que están fabricados con polímeros, de forma que cuando regamos a la planta nos ayudan a observar los microbios. tienen la textura adecuada y unas propiedades químicas específicas, y son inteligentes porque también incluyen sensores que nos permiten conocer dentro del suelo qué cambios se están produciendo. Otra cosa que hacemos, va despacito esto, es que desarrollamos microscopios que nos ayudan a ver las muestras a gran tamaño. Por ejemplo, una técnica que hemos desarrollado, esto es como una capa de luz perpendicular. Es como un láser es como una línea de láser, y colocamos la cámara de forma perpendicular y vemos qué ocurre exactamente en ese plano. Y esto se puede escanear para poder reconstruir todo el volumen en torno a la raíz, y podemos utilizar distintas técnicas para saber dónde están las distintas partículas, microbios, etc. Así que es un poquito difícil de ver en el vídeo. A ver, no sé si se puede ver así. Vale, así. Así se ve mejor. Vale, hay vídeos, no sé si se pueden poner. ¿Vale? Estos son los datos que se obtienen. Obtenemos grandes conjuntos de datos que nos permiten reconstruir la rizosfera in situ, en el suelo no saturado. Esto es un logro tremendo y podemos verlo en directo conforme va creciendo la raíz, y vemos cómo se distribuye el suelo en torno a la raíz. Otra cosa que estamos haciendo es empezar a ver el exudado radicular para ver los anticuerpos, los distintos polisacáridos, de forma que podemos ver dónde están los microbios, dónde se produce la alimentación. Estos son los datos que nos permite recabar esta técnica. También hemos desarrollado lo que conocemos como sin etiqueta, porque en muchas ocasiones necesitamos una etiqueta y tenemos distintas técnicas. Esto es como la luz cuando utilizamos un puntero y la dinámica se relaciona y nos permite ver la actividad biológica que se está produciendo. Y esto nos permite detectar dónde están los nemátodos y cómo responden a los nematicidas, por ejemplo. A la izquierda, por ejemplo, lo tenemos en suspensión simplemente, y podemos ver cómo se mueven rápidamente. Y un poquito menos relevante, pero también muy interesante, una propiedad física muy interesante del suelo es la compactación del suelo, que en algunos casos trabajamos con maquinaria agrícola, etcétera. Hay distintas cámaras que utilizamos para ver cómo las raíces responden a esta compactación. Entonces, ¿qué hacemos con estas técnicas? Os voy a presentar este trabajo que hemos realizado sobre el movimiento de las bacterias. A partir de ahora, lo que os voy a presentar está basado en una cepa que utilizamos en el laboratorio para hacer ensayos, y que utilizamos sobre todo con tomates, lechugas y trigo principalmente. El origen de este trabajo, comenzamos a interesarnos en esta cuestión ya en 2014. Entonces no contábamos con estas tecnologías que acabo de explicar, no podíamos ver los microbios, pero nos interesaba esta cuestión. Así que creamos un modelo matemático. Asumamos que la raíz crece y empieza con ciertos microbios, distintos tipos de microbios en el suelo, pero las raíces van creciendo. Entonces, ¿qué propiedades tiene que tener ese microbio para permanecer en la raíz y colonizarla? Y a partir de distintos aspectos que vemos en la pantalla, los exudados, vemos distintos patrones de colonización y en algunos casos los microbios pueden permanecer en la raíz y en otros no. O se crean patrones que no son realistas. Lo interesante es que vemos que lo que ocurre en la punta de la raíz puede ser bastante importante. Así que a partir de estos resultados podemos utilizar nuestro sistema experimental para empezar a ver qué ocurre en realidad. Porque nadie se lo cree simplemente con modelos. Así que empezamos a utilizar nuestros sistemas. Este es un microscopio muy simple que se puede encontrar en todos los laboratorios y estos son los datos que obtuvimos. Tomamos muestras cada 12 horas. Vemos las bacterias en torno a las raíces y lo que podemos ver es que los patrones son muy distintos dependiendo de si las bacterias son móviles, es decir, según el patrón que crean, vemos que en un lado los patrones son menores, en otros son muy superiores. Y aquí vemos cómo está colocado en cuanto a las partículas. Así que colocamos esta información en un algoritmo que automáticamente nos dice en la imagen que es un biofilm en la raíz y que no lo es. Y luego con esta información construimos mapas espaciales y temporales de dónde se encuentran las bacterias. Y encontramos resultados muy interesantes. No tengo mucho tiempo de detenerme en ellos, pero claramente lo que vemos en la izquierda es que las bacterias no se distribuyen aleatoriamente. A la izquierda tenemos biofilm y a la derecha vemos que no es aleatorio. Las bacterias eligen a dónde ir. Y otra cosa que hemos encontrado es que tenemos un patrón temporal claro. Al principio tenemos bacterias móviles en torno a las raíces. Ese es el punto de contacto de las raíces. Pero no tenemos biofilm en las primeras 24 horas. Después el biofilm empieza a formarse en las zonas más maduras de la raíz y luego las partículas del suelo vemos que están cerca de la raíz, pero no tienen patrones. Están distribuidas uniformemente a lo largo de la raíz. El principal problema de esta situación es que no sabemos dónde están, que estas bacterias son muy heterogéneas. Así que utilizamos un microscopio de luz para tener una resolución temporal.
Y nos sorprendió mucho ver cosas que nunca habíamos visto. Las bacterias funcionan como un grupo, así que forman nubes o como bandadas y funcionan conjuntamente, avanzan de forma muy eficaz y empiezan a formar el biofilm en la raíz. Al estudiar la genética, vemos que está relacionado con la movilidad. Si vemos los genes involucrados, vemos que tenemos patrones que son más difusos y tienen efectos en la colonización de la raíz también. Otros factores como no se vieron afectados, como las frentes difusas y el ramificado. ¿Cuál es el estado si es viscoso o no? Eso también puede afectar el comportamiento. Si es más viscoso, vemos que este patrón es inestable y podré explicarlo un poquito más tarde, estos diagramas, pero el hecho de que hay cosas que no se pueden mantener, la forma no se puede mantener y se desagrega, digamos, y se aporta en otro momento. Mientras que si tenemos algo que no es viscoso, tenemos este comportamiento del grupo de la bacteria que parece bastante estable. Y lo que estamos haciendo ahora es que nos estamos, digamos, rastreando, digamos, las distintas células. Vemos este punto que se ve aquí en esta imagen. Estos puntos son los mismos bacterias que se monitorizan y las hemos puesto todas juntas para que se vean las direcciones y se puede ver que hay corrientes de bacterias en el suelo y también sabemos que estas bacterias afectan a todo, como si fueran corrientes de sangre. Estamos en un proceso en el que desarrollamos técnicas para cuantificar el flujo de bacterias en el suelo y también relacionar cómo estas células se relacionan y cómo se relacionan con la colonización como grupo y colonizan la planta. Para recapitular, esta es mi última diapositiva. He acelerado un poco porque me ha parecido que íbamos con un poquito de retraso. Así que lo que estamos haciendo en el laboratorio es desarrollar la técnica para resolver la actividad microbiana en la rizosfera del suelo. Y esto es muy importante porque, como espero que hayan visto, estamos empezando a ver el nivel de complejidad de los mecanismos para colonizar la raíz. No es algo sencillo que se difunda así tal cual y de alguna manera está cambiando los modelos que teníamos en mente sobre cómo se dispersan los microbios. Hay que tener en cuenta que no es la típica dispersión que solemos ver, por lo menos para algunas bacterias. Espero que haberles convencido de que el movimiento es crítico. Una vez más, cuando se introduce una bacteria, cuando se introduce una semilla en el suelo, no tiene microbioma rizosfera. Si lo tiene, no tiene nada que ver con el que tendría en este otro caso. Así que entender este establecimiento, lo que se necesita para construir alrededor de la raíz, para construir un entorno saludable, hace falta movimiento. Puede ser pasivo, pero también puede ser muy complejo. Y quizá es una característica que debemos buscar para introducir una nueva cepa en el suelo. Lo otro que estamos empezando a ver que el reglamento genético de este proceso hay que tenerlo en cuenta y este es el principio. No entendemos del todo. Y otra cosa es que no hay que olvidar que el entorno físico tiene efectos muy fuertes. Si nos fijamos en la bibliografía y nos remontamos al movimiento de bacterias en el suelo, lo primero que vemos es el contenido del agua, que es un motor principal de colonización. Si no hay agua, se mueven muy poco y si hay más agua, entonces van a hacer muchas más cosas. Así que la diferencia en estos términos es muy importante porque si nos fijamos en la literatura o la bibliografía, vemos la importancia, vemos información importante relacionada con la resolución de este problema. Muchas gracias y si tienen preguntas, por favor, no duden en hacérmelas. Muchas gracias.
Thank you very much, Lionel. Thank you very much for sticking to the time. Now we're welcoming Jose Nolasco from Trade Corp, who's going to talk about biostimulation of concentrated power, a key tool for quality and performance in crops.
Good morning. My presentation is meant to be up there on the screen. Yes, perfect. There it is. Good morning, everybody. Before I start, I'd like to thank you for giving me the opportunity of participating. This is my second time. Thank you so much. It's a pleasure and an honor to be here with you. And secondly, I'd like to congratulate you because this is amazing. There are about 1,000 people here. This is not normal. I mean, well, it's normal for you because it's the third time you do something like this, but it's not something common to have this kind of symposium that are that successful around the microbiome. So congratulations on the work you're carrying out. My presentation is related with biostimulation of concentrated power, and that in small doses, 50, 100 milliliters per hectare, is capable to have powerful effects in the quality and yield of crops. This is what I'm going to talk about today in my talk. I always think that in this kind of meetings or gatherings, we have to make sure that we exchange our contact details, and this is key. Even as important as the information we can share today. I'm José Nolasco, head of Global R&D Bionutrition, and I'm also a lecturer in the Technical University of Madrid. You have my email address, you have my telephone number, so that you can contact with me if you deem it necessary or appropriate. Whenever I talk about this, I always start with this slide. I've used it about like 1,000 times, but it's very interesting as an introduction. In general terms, all have attached different boxes of knowledge, different branches of knowledge. The nutrients, protection against what? Pests and diseases, everything related to the application of water and irrigation. That is very relevant. The fourth leg is inevitable. The fourth leg of today for sustainable agriculture, for the future of agriculture, is the management of stress. This is the way it is. Without this fourth leg, we're not going to move forward. Does anybody know? Or has anybody thought about stop watering, stop irrigation? Well, I'm not going to contribute nutrients. Let my crop die with the attack of pests. Is someone not thinking of fighting abiotic stress? Well, this is in quality and yield, because the farmer wants taste, color. What they want is profitability. This is what biostimulation is carrying out, and this is all interconnected with everything that enables agriculture to create wealth. I've talked about biostimulation in many fora, and I always say the same thing. I'm going to repeat it here. Biostimulation is never cost. It's just investment. That's it. Full stop. Investment without any doubt. And it's not, we don't have to think of it as cost, because the impact in what is really important is not kilograms per hectare, but euros per hectare is related to quality. And this is what biostimulation does, without any doubt. And this is the role that this technology carries out. I have 15 minutes, but I know that there's a coffee break afterwards, so maybe I can get an extra minute. You look like you're good, and you'll probably give me some more minutes. But if biostimulation is something fundamental, how do you design a highly efficient biostimulant? In the orbit of nutrition, we talk about mixing components. I have potassium nitrate, I have magnesium sulfate, and I have them all together, and there's a synergy. I have a solution. I'm happy. Thank you. Excellent. You're happy? But if I do that in biostimulation, the problem is enormous. If I mix substances that have a clear effect against osmotic stress, and I do it thinking that I have the best solution ever, interactions among them are going to be brutal, especially if they act in similar ways. Anybody that wants to design highly efficient biostimulants for fermentation has to follow a mode of action. This is the way to go. And you'll say, okay, yes, well, there are different labels that say that the product does everything, but that's a lie. The action mode is fundamental and critical in the development of a highly efficient biostimulant. No, you have to know what it doesn't do and what it does. And this is the only way to proceed. And this is what we do in our company. And the only way to achieve results with 50 or 100 milliliters per hectare is based on this, on the phenotype. What happens to the plant? Does it have a root? Are there any flowers? Is it growing more? Is there development? What is happening with the plant? I mean, you can get this with scientific data in agronomic trials, but you cannot stop here. You cannot stop here. This is connected with the metabolomics of the plant. If you can improve the fattening or the growth of a plant, there are things that are increasing in the plants. This metabolomics is essential, and otherwise you don't know what's happening with the product. And this is connected with the genetic response, the transcriptome. What are the roots that are activated and which ones are the ones that aren't? What genes have been overexpressed or underexpressed? If you say that a gene is turned on and off, please say that you cannot do that. A gene is not turned on or off. I've seen it in many conferences, but that's wrong. You can express it or not. You cannot turn it on or off. You can do that with the light, but not with the gene. That was just a comment. Side comment. Transcriptome and the phenotype and the health of the plant is something that you need to take into account. And this is something that we've done with this in this situation. We've worked with a microorganism that yesterday, today, or in the future, you'll have had in any meal with an enhancer of flavor. There are many strains. We've isolated just one that is capable of carrying out this fingerprint. More than 250 metabolites, primary and secondary metabolites. Two, with fermentation in a specific substrate. Circular economy, coming from sugarcane in factories in Brazil, and what changes is the substrate, the proteins, the metabolomic profile is changing with a specific process involving fermentation, CO2, level of oxygen, timeline. Whatever you change, it has an effect. So here we have microbiological fermentation, and this is what enables the product to do what I'm going to talk about right now. With more than 250 agronomic trials, with intense R&D, being capable of handing in these results with 100 milliliters per hectare, with concentrations, this is all black, I don't know why, with a single stamp, with a single metabolic profile. We have done this at European level, guaranteeing the effectiveness and any aspect relating to if we take into account the agronomic characteristics of development connected with what is the genetic expression of the crop when we deal with these metabolites, what are the metabolite pathways, the genes that have been overexpressed or underexpressed, together with the metabolomics of the plant. We've done it with the University of Milano, and this is what I'm talking about. And if we want to increase the development, what you've put in practice is photosynthesis, translocation, primary metabolite, and this is what's happening. Otherwise, you're lying. These are the main three pillars in a technological development. This is what we work with. We've been working for a while now in the microbiological fermentation project. when you increase the precociousness, the weight of fruits with application of 100 milliliters per hectare, when you see that you're capable of improving the fruit, the mean or the fruit average weight, the number of fruits, the total yield, or the juice content, when you see that you can increase the precociousness or the number of fruits, the yield with application of 100 milliliters per hectare, that's because you know that what's happening to the plant once you've applied it. When I show this slide, you'll probably say, what does this 61 mean here? These are genes that are overexpressed or underexpressed. What is this 61 here? This is just modulation. But how do I translate this? What is technology doing? The technology, I don't know which one is mine here. Okay, anyway, the technology that we are developing synthesis of the cell wall and the photosynthetic activity and potassium activity. What I do want is the development of the fruit, photosynthesis. What do I need? The production of sugars, yes, of course, and translocation of sugars, yes, of course. It's important the role of potassium. If I manage to get this in the plant, this is the result of the product, and this is the summary of the transcriptomy. We managed to prove that with over 250 metabolites, that primary and secondary metabolites in the product, we managed to activate photosynthesis, the production of sugars, and the mobilization of sugar. Let me show you some images that we took with a professor from the University of Madrid, Victoria Fernandez. On the bottom image, it looks like there was an atomic bomb laid there. What happened? There are things that have been completely destroyed. Photosynthesis. This is because of the drought. This is due to the lack of water. These leaves are not going to carry out their job, and the impact is obvious. When you take the same images, but with the solution of microbiological fermentation, the structure is there. The mesophil cells are there. And this is completely linked to the previous slide. Synthesis of cell walls, membrane structure, cell division. When you see this in a picture, you can die. I mean, and without any kind of hormone. When you talk about products with 100 milliliters per hectare with obvious effects, well, you say here there are hormones. No hormones whatsoever. Why is it capable of doing this in these low doses? Owing to the 250 primary and secondary metabolites that we manage to the microorganism to have with this extract, with this strain, can be produced. This single fingerprint profile in the product, this enables the product to do this with a clear effect in quality and in development and in quality of fruit. 2 minutes, Jose. Perfect. I'm going to take another 2 maybe. That's a lot. Just twice as much. The analysis of the tomato metabolic profile, for instance, what is primary primary metabolism? Synthesis of amino acids. That means survival. And the secondary one is its ability to adapt to the environment. If I take a look at the metabolomics, what are the metabolites that appear? Photosynthesis, sugar related to the different sugars the mobilization of sugar, and this is what you need for the development and the quality of fruit. We're talking about amino acids, organic acids, sugars. I only have 2 slides left. The application of these products have a direct effect, and this is not what I'm saying it myself because I want to. The metabolism of these carbohydrates, the application of quickcorn has a direct effect on the metabolism of carbohydrates. This is telling me exactly what the product does. And this is then there is an image on the field that this is the agronomic trials of scientific nature. 2, the treatments with quickcorn have an influence on the photosynthetic activity and in the signaling, with an effect on the growth or development. Last slide. A specific bacterial strain. You're experts. I'm not going to tell you the difference between one strain and the other. One thing is very different to the other. We have to identify exactly the one we need to produce the metabolites that I need and the concentration that I want. The metabolomic profile of the product. 2, sustainable source of natural food with proteins and sugars needed. Circular economy because it's coming from the factory. And 3, with an exclusive biosynthesis process when you control CO2, time. These 3 things enable you to contribute to take our small step in bio innovation and in productive in products that I hope I have managed to explain in this talk. Thank you very much. Thank you, Jose. You know that the questions have to be asked through the app, and all speakers are going to reply through the app. And now we're going to have a coffee and networking break, and we'll be back in 20 minutes.
Good morning to those of you here at the auditorium and those of you online. Now we're moving to the round table that's sponsored by Info and is led by Antonio Romero. And to follow with the nice presentations we had before, here we have with us Pablo González Conejero. He's the chef of the restaurant Cabaña Buenavista that has 2 Michelin stars. And we also have Joaquín Hernández, who's the general director of BSI Wineries. Thank you very much. First of all, I want to welcome all of you to this second part after the coffee break. And first of all, I want to congratulate the organizers. The Microbiome Conference has around 1,200 attendees, both on-site and online. So I think we have to congratulate the organizers for organizing an event that is so important for our sector. I'm sorry, but the General Director of Info wasn't able to come here this morning. He would have liked to be here with all the guests that I'm going to introduce now. Briefly, for those of you who do not know us, we are the Development Institute of the region of Murcia. Ours is a body that facilitates innovation for businesses in the region of Murcia. We are well known because we manage funds from the EU, from FEDER funds, and other funds that are not just financial funds. We're working on training. We help companies to develop procedures for innovation. And we work in companies at workshops. In case you're interested, please do not hesitate to let us know. We are also helping many businesses positioning themselves abroad. And there's a network of technological centers to bring innovation processes closer to businesses in the region. I don't want to take any further in introducing the development agency. You can contact us via our website. I work at the Innovation and Digital Transformation Department. So you can contact me or any of my colleagues. We are at your disposal. Today we had a challenge, and it was within this conference, try to talk about innovation, but in a different way. And we wanted to have 2 guests, 2 speakers who lead different businesses to have a dialogue. In this case, Bodegas San Isidro Wineries, and the restaurant Cabaña Buenavista. Why? Because they are producers and users of agricultural products. And they use technologies that are going to be mentioned in this conference that enable us to produce more sustainable products that can stand a higher water stress. And afterwards, if you have to cook those products, as Pablo González does, of course, the product has to be in the best conditions to get a spectacular dish. So first of all, I'm going to introduce them. I'm going to explain some aspects of their careers. Pablo González Conejero, I think you all know him. He's very famous at a regional level. He owns 2 Michelin stars, and He was the owner of the first restaurant in the region of Murcia to get the first Michelin star. In 2004, that's a long time, he opened Restaurante La Cabaña, and he has received many different awards. Apart from working at the region of Murcia, Pablo also helps, is a consultant for other restaurants, such as the restaurant of Hotel La Boadilla, with a Michelin star in the province of Granada. He has worked at Arsac restaurant, and he is always willing to learn, to show that learning in what he cooks, always innovating in design, in flavor, to offer the best experience to those visiting his restaurant. Apart from the 2 Michelin stars, he got 3 Repsol Suns, an award by the International Academy of Gastronomy, and in 2018, he was acknowledged as the Person of Murcia of the Year. And Joaquín Hernández Muñoz, he's the General Director, the CEO, and the President of Bodega San Isidro. It is a cooperative that's 90 years old. It was created in 1934. And we wanted him to be here today because he innovates in his company in the 70s. He moved from bulk to individual bottles, and they have been innovating for many years, as he's going to explain. They are really devoted to research. They collaborate with different research bodies, and they have developed different projects with public and private organizations. And Bodega San Isidro, they have received many different awards in different countries where they sell their wines. And he has been working for more than 30 years as an example for all cooperatives in Spain. He was the president for 12 years of the Association of Cooperatives of Wine in Spain, and he's the president of the Economy Circle. He's the chair of the Circle of Economy of Murcia. He is also working in aspects related to biotechnology, and his main raw material is the local grape Monastrell. Good morning to both of you. I could spend more time talking about your CVs, but I think we should start our discussion. My first question is a question for both of you. Pablo, maybe you can start. What's the role of innovation, reinventing for a company to reinvent itself constantly? Good morning to all of you. I think we are all aware on the importance of innovation. We work in a sector that doesn't give you time to enjoy what you're doing, because at the moment you have finished, you have already started thinking of what you're going to be doing tomorrow. There are many people who ask me, why don't you remember dishes from the past? Why don't you create a menu with historical dishes? And I don't like that idea at all. Because if I look back, what I did in the past, wouldn't have any sense today, because it is necessary to be constantly innovating. So I would say that for us, having an R&D department is essential. Our company, our restaurant has an R&D department that reflects our purpose, our goal. Because the speed out there in the street doesn't let you to stop paying attention to innovation. We have to put ourselves on the customers or the client's shoes, and they're the ones who want to enjoy the experience. And what about Bodega San Isidro? What are you working at regarding innovation? Good morning to all of you. Thank you to the organizers for organizing this conference in Murcia. And I think the organizers have to choose these dates, because as you know, in Murcia it doesn't rain a lot, but in the last few days we have had many storms and lots of rain. Bodega San Isidro, our commercial brand is BSI, was born in 1934 in a complicated period in the Spanish history, and since the end of the 50s, beginning of the 60s, it became a national referent due to the wine and its features. In the 70s, we realized that once we were going through a crisis, we asked ourselves, what can we do? And the decision which was taken was that there was a local grape variety, that's Monastrell, and it had been used in Jumilla for more than 3,000 years. This grape variety was there for a reason, because in the region of Murcia, which is very dry, the southern part is even drier than the Sahara Desert. And also our area is very dry. So at that moment, we said, we don't know if this is going to be useful in the future. We're talking about a period when many different wineries started closing down. And what we did was to start researching. We reached an agreement with Dr. Francis from the University of Bordeaux. He visited the region of Murcia, and he said, you had a very big potential, but there were things that had to be changed to make the most of that lack of water. So we started researching on different cultivation methods, how to optimize irrigation, collaborating with the University of Murcia, the University of Bordeaux, the University of Castilla-La Mancha, and more than 200 projects and PhDs have been developed. Today, in the last years, many different projects have been developed. At the moment, we're taking part in 6 different projects, and we collaborate with different entities and universities. So from the results of the research projects, nowadays there are wineries that we started collaborating with, and we got to take Monastrell variety to be one of the best known in the world. In the USA and in the UK, Monastrell variety is highly appreciated. Innovation has been key. First, it started with the crop, then with the processing, and then the presentation. So innovation nowadays doesn't have anything to do with what it was in the past. We have to go on changing, and we have to see what pieces of research we're going to carry out to avoid water stress, but we also have to think on health, on future consumers. They need new sensations, new feelings, and they are looking for a quality product. And talking about consumers and what they need, in the case of Pablo, he offers customers what they want, what they demand. And in order to do so, he uses the production of regional companies to prepare and to cook his dishes. So what would you request producers? What can they improve for you to potentiate flavors? What things can be improved? I think that the most important thing is that we are chefs, we are not magicians. Our work starts where these gentlemen are. If they're not there, we have no raw materials. So we are looking for the best product, the best quality. And how do we get that? For me, farmers are doing a really difficult job because they don't know what's going to happen tomorrow. We can see that in Murcia it doesn't rain, but when it rains too much. So for farmers working in the field, they have to look at the sky day after day because they know that something can happen and all their work is going to be wasted. The same happens to the people working with wine. When August arrives, they're waiting for the day when grapes can be harvested. Because if it rains heavily, all their work can be wasted. So farmers, for me, are like superheroes because they're doing a job that doesn't depend on them or on anyone. It depends on climate conditions, weather conditions. But that's why we need sustainability. There's something that's very important. Murcia, and in this case Jumilla, and the Monastrell grape variety, this was that easy and that difficult at the same time. We decided to start using different grape varieties that do not have anything to do with our climate, with our environment. If Monastrell works perfectly well in our area, why do we want to do different things? Let's make the most of the Monastrell variety. And as I'm referring to this grape variety, we can refer to any other crop variety. And I ask farmers to understand me as I understand them. I want them to come to our restaurant. I want them to see how much we need their effort, how much we need their innovation. We have been collaborating with IMIDA for a long time in the recovery of autochthonous and local varieties. We want to have the product that grows perfectly well in its environment. We don't want products that come from different areas. We want local products, and that's what we're working on. How do we grow hand in hand with farmers? What we're doing now is to go to the field from the moment of the planting, and we follow the whole growth. I'm going to mention green beans. We've got many examples, but at the moment we're working with green beans. They have just been planted, we're at the season, and the farmer looked at us like saying, "Are you crazy?" We had tested different varieties in the plot tests of IMIDA. If we get a green bean of 1 centimeter, for you to have an idea of what I'm talking about, that green bean that's amazing because it contains the whole flavor of a long green bean, but in a tiny space. You don't even have to cook it. We don't like to cook it. We like to use it raw. But that's a product with an amazing taste, that's crunchy, but it also has a flour that tastes of green bean, and the taste is amazing too. And when we go with people from IMIDA to the field, they make fun because we are eating flowers, weeds, plants, not fruits. But they have to realize what that small product can transmit. And we need farmers to understand that we need to work hand in hand with them, and we need to collaborate with them, and we need them to understand us. We need to grow with them, and we need them to understand us. And now we are collecting 1 centimeter long green beans that are amazing. That's what possibility, what innovation is giving us. That's the possibility that's giving us. The same happens with the Monastrell grape variety. Why do you start using different varieties that come from different areas that do not have anything to do? In Spain, terrible things have been done, such as working ice wine in a freezer. Ice wine is done in Hungary because the grape freezes because of the cold. And in Spain, some people decided to freeze the grapes to produce their wine. Can you imagine it? So that's something we need to fight for. And people need to understand that our collaboration is essential. The performance you get when you work with local products cannot be compared with any other product. And in this edition 2023, attention is paid to companies to adapt their production systems to the demand so that the perception of clients is better, such as regarding nutrition, environmental footprint, etc. The reduction of the use of supplies. Joaquin, what are the challenges that you are developing regarding new varieties, sustainable practices, techniques to improve grapes after harvest, or the production of biological agriculture products? I think we all know that. And today we are at a forum of specialists who work for that. At the winery, where we have wine and oil, almonds, but wine is our main product. But with oil, we are also working with the local variety. we always say the same, and it's that if you don't have a quality product, you're not going to have a good product, and chefs or people at their houses will not be happy with what they get. So we have to convey the importance of quality to producers, to farmers. Pablo was saying that producers in the past thought that in the case of green beans, the longer the better, but that's not the case at all. And in our case, in the winery, I think we were collaborating with Bodegas Torres. We did in the 80s a quality selection, because I don't know if you know that the quality of wine changes depending on the kind of grapes you're using, where they're planted, how the plot is oriented, etc. So we have a team of experts who did a technological selection process that we developed together with Bodegas Torres from Catalonia, and this has been implemented in many other places. What are we doing at the moment? At that moment, we saw that apart from the Monastrell grape variety, there were some grapes, vineyards that supported some conditions better than others. We were the first ones to sell a specific vineyard. I don't know if you're aware of Xylofera. That's a disease that affected many different areas in Europe. And some vineyards were healthy, but the European Union came and said that that couldn't be used. So we kept that vineyard and we saw how that could be used. We also saw that biological crops were essential. But we saw it because there wasn't anything else we could do, because we didn't have water. We weren't able to use many treatments. So we were the first world producer of ecological or biological wine. Sometimes we wrote biological agriculture on the label. It was tasted by experts and they said, no, this is not tasty. But now the market has changed, and those wines, if they're properly elaborated, they constitute an advantage. It's like wine without sulfates. We started creating wine without sulfites because there are people who may be allergic to it. The first market was Japan. I thought it was going to be difficult, but now we are the leader of wine without sulfites in the world. In Murcia, we have a wonderful weather for local varieties. Pablo is more well known outside Murcia than in Murcia. He's a star, but not a Michelin star. He's a star outside Murcia. And other producers, the same happens with other producers. So what we're doing is we're going to go on selecting products, and we're going to meet the customer needs to make a selection of products of wine, grapes, olives, oil, almonds, to create some niche. If you go to a country that is more keen on sweeter wines, we create a sweeter wine. If we go to the center of Europe, who like more drier wines, we offer them drier wines. And that's why we need to help producers help us too. And of course, farmers, they need to be profitable. Their productions need to be profitable. Yesterday, I spoke to a producer from Murcia who was crying because he had lost in 2 days the effort of many months because of the rain. So we need to see how we can adapt to consumers, to chefs, to the land, to get a more profitable agricultural production and to get more youngsters to work in the world of agriculture. Because AI has been invented, but a world without food hasn't been invented yet. So I think this conference is essential because most of you are technicians and the sector is doing a great job for that to develop and for it to be profitable for all of us and to adapt to the different types of consumers. We are aware that we are lagging behind, so I'm just going to ask 2 very short questions and we're going to finish. Pablo, have you seen that your clients perceive in what you cook the use of local products? Or that after the cooking, they do not perceive those initial properties? In this case, I'm referring to products from the region. In the last 20 years, we've been working, we've done many different things. We are also vulnerable to market changes. And some time ago, there was a period that was featured by globalization. We got products from many different places, and we started working on that sense. Until a few months ago, we decided to stop. Why? Because we saw that when we referred to local products, when we refer to our culture, the dish was better perceived by our customers. We're not talking about a restaurant where you go to have lunch. It's a restaurant where you go to get feelings. So nowadays I can say that the menu we have just finished is fully under the umbrella of the region of Murcia, from the beginning till the end. We called city and town councils. We talked to ladies in little villages. We talked to Alfonso, who has a program, a TV program, Loco Mio, I think, who visits all the villages gathering information. Because the aim was to recover our culture, to recover concepts, and to highlight what we are or what we were. We created the Busca salad, recovering different wild products and different herbs that I wasn't familiar with at all. But they're part of our culture. This salad is what people prepared with weeds they got from the fields because they had nothing else to eat. Lios from Lorca. This is lamb that is cooked in the oven and it's slowly grilled. We are working thanks to the fact that we have collaborated with city and town councils, with Alfonso, in order to look for products, to look for our roots, to interpret it on our way. There's a wonderful story. We have recovered a specific type of olives and we reinterpreted them. The ones that were left on the floor, poor people harvested them and they used them. All of these stories can be found at a restaurant. And in fact, the menu is called Region of Murcia because it is made up of concepts from the region of Murcia. The salty fish, different types of cooking, pickled vegetables. There are people who come to the restaurant and cry because flavors bring memories. Maybe you can remember your grandmother, your grandfather, your aunt, your uncle. And it is wonderful for a person from the area because it brings memories, and from people from outside the city and the region because they get to know our culture. And as the final message, Joaquin, I would like you to convey the importance of investing on innovation, of collaborating with universities, technological centers, how important it is for a company such as yours to work with innovation. All of the public and private collaborations are essential, as I already mentioned. But I wanted to convey a message that Pablo has already mentioned. As the president of the Circle of Economy of the region of Murcia, I heard a person saying that we need to highlight our features as a region. And I also heard him saying that innovation has to be based on our products, on the customer needs, but we also have to take into account, and that's something that always comes to my mind, and I think this is very positive for agriculture and for technology. Nowadays, consumers and our society and the European society, fortunately, we are able to eat and drink every single day. Our basic needs are covered. But youngsters nowadays are demanding new products. They want functional products. They want local products. They want sustainable products. Some years ago, we talked about globalization, but youngsters nowadays want environment to be respected. They want sustainable products. I was surprised to see youngsters in the city center of Murcia with posters saying, let's save the planet. But this morning, I was listening to the news and I heard that we had the warmest spring ever. And the temperature has risen 1.8 degrees centigrade. So we have to think in a healthy society, in the future, in sustainability. So the future is based on functional, profitable products for the whole production chain that can be well received by consumers. Because this is not just a matter of cooking nicely, of using nice labels. I think all of us need to get people to be moved by our products. Let's imagine that nowadays we don't talk about milk anymore when we see advertisements. Now the advertisements talk about the package and that it's sustainable, etc. Why? Because the company knows what consumers need. And I think it is a very good moment. And as a person from Murcia, the problem is that we think we are ashamed of acknowledging our values, of promoting our businesses, our success. Some years ago, I introduced my company and I was said, no, that's not the way to do so. You need to be proud of your company. You have to be proud of where you come from. We are a region where we cultivate with just a little water, where we cultivate high quality products. And here we are producing things that cannot be produced anywhere else. So we really have to be very proud of our products. Sometimes I think it's a shame that our products are better valued outside Murcia than inside Murcia. It's really a shame. Okay, so those are the final messages, both from Pablo and Joaquin, encouraging us. I think positioning our regional products have to encourage us to innovate even more. And we need the technology for a more sustainable production. And conferences such as this one enable the collaboration between the public sector and private companies. Thank you very much for being here today, Pablo and Joaquin, for this talk. And thanks to the audience for your attention. Thank you very much.
Thank you very much to our speakers in the round table. It was a pleasure to have you here. Now we're going to continue with our papers. We could be here with Pablo and Joaquin forever, but we need to move on. And now we're going to give the floor to José Antonio Rojas from Servalesa. He's going to talk about the effect of application of consortia of microorganisms in the soil and polyvalence aspects. I'd like to remind you that you can ask questions through the app. The speakers will reply later. You'll have the chance to have a look at the answers they provide. Jose Antonio is ready. Good morning, everybody. Everybody here in the room and all of you following us online. Thank you for your introduction. In these 15 minutes, you're going to hear the word polyvalence or multifaceted for many times, and you're going to see why it is important. What I want to show with my paper is the need that we have lots of microorganisms in the soil to get to know the traits and the advantages they bring. For many centuries, we know that soil is essential for life because the plants eat from it. This is not nothing new for all of us here today. However, it is now when we admit that life is essential for the soil. Without the microorganisms that live in it, it would be impossible to have biochemical cycles and the decomposition of organic matter. For many decades, with the production of phytosanitary products, we have erupted in the soil and we have interrupted the geochemical cycles of nitrogen and other elements. On the one hand, industrial processes have increased the population in the world, and we have lost biodiversity and degraded the ecosystems, agricultural ecosystems. This is disappointing because this kind of management was ideal, and the results were optimal very recently. As I was saying, the results were optimum. Sustainability. With sustainability, what we want to do is respond to the needs of the present without compromising the resources of future generations. We all agree that sustainability is a demand of society. In this global and environmental situation, science has diagnosed that there is a problem. But in turn, science provides us with a solution. This solution is called biomimicry. And why is biomimicry? Something very simple. It's just the imitation of natural processes for the resolution of technical problems. I think this is going too fast. Can I not go back? Okay. Do we have, can we please help? The result of applying natural processes for the solving of problems. Excuse me. Okay. So in agronomic terms, a holobiont is the system in a plant that works as a hosting organism, and the microorganisms that have a mutualist relationship with them. In the rhizosphere, there are many microorganisms, fungi and bacteria that have a relationship that is a mutualist relationship. That is to say, microorganisms feel attracted by the secretions issued by the root system of the plant that are rich in carbonated compounds coming from photosynthesis. In this way, the plant kingdom wishes to see what vegetation is going to have around. The result of applying biomimicry in agriculture, replicating the diversity processes, is called consortium of microorganisms. We're not alone in this line of work. For me, it's important to see that many people are working on this line of work. In mid-April, a researcher from Granada published a paper about the effects of consortium of microorganism in an oak of high ecological value. The microorganism consortium provides advantages that normally come from diversity, adaptability, multi-usage or polyvalence, and synergy. The adaptability is the ability to adapt to develop. Multi-usage or polyvalence is the ability to play different roles, and synergy is the property that reveals that effects of consortium are better than all of the strains that compose it independently. What the systems need of a good consortium of microorganisms, we need the main things that we need. We need for it to provide phytohormones and biocontrol. We need a consortium with biofilm and elliptic enzymes, and the main abilities of fertilizers is that they decompose organic matter, fixate nitrogen, solubilize phosphorus, solubilize potassium. With all these conditions, the best consortium of microorganisms is the one that is most multi-usage one, that can develop more roles. If we had an analysis team or equipment, if we introduce a strain of microorganisms, it's the good thing is the abilities that each strain can develop. Well, have a look and what happens. If we include a strain of Bacillus subtilis, we see that it's got high capability of biocontrol, and in biofertilization, it's capable to solubilize some phosphorus and potassium. If we analyze a strain of Bacillus prumulus with good biostimulant abilities, biocontrol is highlighted. In biofertilization, it can produce siderophores. With the analysis of Bacillus siamensis, we can produce phytohormones, and in biofertilization, it can only provide siderophores. Let's see what happens when we analyze Bacillus amyloliquefaciens. High biostimulation abilities, also biocontrol abilities, but in biofertilization, only decomposure of organic matter. In the analysis of Bacillus megatherium, we can see good biostimulant capacities. Biofertilization, we highlight good solubilization of phosphorus, potassium, and producer of siderophores, but we see that in biocontrol it's not very good. Finally, the analysis of Pseudomonas fluorescens shows good biostimulant capacities, biofilm, light-thick enzymes, solubilizing phosphorus, potassium, and production of siderophores. What happens if we formulate a consortium of microorganisms with all these strains that we meet the requirements demanded of biodiversity, adaptability, multi-usage, and synergy? Okay. So far, we managed to design a consortium of microorganisms with 6 strains that meet all the requirements that we've already mentioned. Now, I'm going to show the results that have been analyzed of some of the studies performed with a consortium of microorganisms. To deepen in the knowledge of biofertilizing components, we've seen that Pseudomonas fluorescens on in the left, 15 days after inoculation, we see the solubilization degree. This same strain of Pseudomonas can solubilize potassium. Only 25 hours after inoculation, we see the first results, and 15 days later, solubilization was at its maximum. We also studied Bacillus megatherium, and after 15 days after inoculation, it stabilizes potassium as well, like the two previous ones. To know the biostimulation capabilities, we analyzed different crops. Before showing figures, I'm going to say that an image is worth 1,000 words. There was a study with cherry tomatoes in Granada, and we see that comparing the same number of fruits that have been harvested in similar situations, the differences are important between a control sample and the treated sample. In this same study, we measured the caliber. Here, there's only a graphic here, but we had a look at the caliber throughout the procedure. With the consortium of microorganisms, the caliber is increased. In this case, the caliber is determinant because it's very important. In this same study, we measured the average weight of the tomato. Here we see some results that are very significant, with an average weight of about 3 grams. What are we saying here? That tomato is not void. Some treatments, you know, that the tomato increases the caliber, but not inside, it's not full. So we created a biostimulation. The biostimulation process has to reach or lead to an enhancement in production or in yield. In our daily work, we're used to seeing how pests of nematodes produce losses in crops, especially in garlic, because it affects the bulb. It attacks the commercial side or part. In these images, on the left, we see a bundle of healthy garlic with everything, with all the layers that are perfect. In the head, we see areas that there is no skin, and the head is completely destroyed on the right. 2.5 minutes left. When we apply a consortium of microorganisms in a plot with nematodes in the past, we see the incidence from 4% in control, and then when treated, we see the changes on the screen. When we see the results that we obtained so far, we decided to study a plot of citrus products. In this case, I have to say that the basic treatment with trichoderma was added a treatment with consortium of microorganisms. As you can see, from the beginning of the development of the fruit, we saw that the caliber was changing and that something was happening with the crop. 2 minutes. In this study, we see that the percentage of foliar cover was mentioned. We see that between the periods there is the absolute increase of foliar cover, and seeing that in September we had an increase of the foliar cover percentage of 4%. As you know, one of the symptoms of this problem is the lack of leaves in the tree, of phytospora. Also, periodically, we measured the caliber of the fruit, and we saw that from the beginning of the data collection until the end in January, when the development process finished, the caliber of the treated sample with the consortium of microorganisms, it was bigger, it was higher. As a result of this work, we obtained a final production of a bit under 43,000 kilograms, and in the control one, and 51,000 of that treated with a consortium of microorganisms. Applying the solution of biomimicry, taking into account sustainability and conserving the parameters demanded by biodiversity, adaptability, multi-usage, and synergy, Cervales has developed and markets the consortium of microorganisms that is more complete with 6 strains. So this was my presentation. I hope I stuck to my time. I just wanted to thank the consortium of colleagues that made it possible for me to give this, present this paper. Thank you very much.
Thank you very much, José Antonio. Now we're moving to the next speaker, Pedro Palazón from Ideagro, who's going to talk about soil enzymes, a key factor in sustainable agricultural production. Good morning. Thank you. I want to start thanking you all. This is the 3rd edition of Microbiome. What in 2019 started as a crazy idea, as Carlos mentioned before. We are all crazy, but we are all together in this crazy adventure. For those of you who do not know me, I'm the CEO of Ideagro. My name is Pedro Palazón. We have been working for more than 11 years developing biostimulants based on microbials. We are a multidisciplinary team with people from many different areas. This is great until the moment we sit down to talk about a project, because we all have completely different points of view. But in the end, it seems to be enriching for the project. We have different types of labs to support all the projects we work in. We are aware of our limitations, and we work to get the best for society. We collaborate with the University of Córdoba, Salamanca, Alicante, Albacete, and we are expanding internationally to Ireland, the US, and Brazil. Something that has always worried us is the knowledge transfer of knowledge. It's great to have new content, but it has to be sent, it has to get to the end user. That's why we work with technicians, with farmers, etc. That's how Microbiome was generated or was created in 2019. And when we started organizing this event many years ago, I thought, how did we get here? How do we have to move forward? And a week ago, I started thinking about it. And I said, we have more than 100 papers or presentations talking about microorganisms. And we always talk about a product, what we expect from it, and what can happen in the future. But there was something missing. No one has ever talked about what is necessary to get a product based on microbials, or microorganisms, sorry. So I phoned Carlos, We have talked about different aspects in previous editions. That's why I wanted to talk about soil enzymes. This is a link in case you're interested in the topic. What I'm going to talk about is how to obtain a new microorganism for agricultural use. Now my time starts. We're always talking about the final product, but how we get there. We always talk about the FAO. It says that the soil is full of microorganisms, and we have to select one. From those microorganisms, we have to select one that we're going to put into a bag, that we're going to apply to the field, and that has to work. So the selection process is really, really important. Last week we were in Barcelona. We are expanding our team, and I started working with headhunters. And they said the first thing you have to do is to follow a strict process to find the best profile, etc. And that's what you have to do, what you want to do, what your needs are. Then we're going to select candidates. We're going to carry out on face-to-face interviews, telephone interviews, online interviews. We're going to do a final assessment. Then we're going to select one and we're going to hire that person. And I said, well, that's the same thing we do with microorganisms. So maybe this is a good idea to follow that process. And let's see the similarities that are there when we want to add or to incorporate a new person to our team, and when we want to incorporate a new microorganism. Thinking that this is a long and expensive process. First of all, what is the product we want to add to our catalog? Do we want a biofertilizer, a biostimulant, biocontrol, bioremediator? Secondly, where can I get these organisms from? From the soil, from the water, from the plant? If they're endophytes, if they come from an insect, from the air? Because we cannot forget about microbiome. That includes everything: the roots, the soil, the phyllosphere, the air. And then we'll get the resources to find our candidate. That would be like microbiome would be the equivalent to LinkedIn. Then we're going to isolate microorganisms. And in order to do so, we're going to do a selection. The position I want to cover is biofertilizer. What are the requirements? It has to work on a pH range from 4 to 8 in temperatures between 5 to 30 degrees centigrade. It depends on where I have to work, where it has to work on. Is it going to be foliar? Is it going to be on the soil? Is it going to work on a specific type of crop? As Carlos mentioned this morning, it is impossible to have a microorganism that is available for all types of crops. And once we decide that, we start isolating the microorganism, depending on where it comes from. We isolate it. And with your cultivation means, you're going to get the extraction, because you cannot imagine when you take some microorganisms on a dish, all the things you get. So with all the microorganisms we get, sometimes it's more than 100, you need to characterize them. You need to find the species, and you cannot imagine how many pathogens we can find Those are the ones that better resist temperatures, pH range. And what we do is to eliminate out of all of the kingdoms. I'm going to eliminate the ones that do not speak in languages, those who do not have availability, etc., etc. So now we have pre-selected some candidates. We have received their CVs. And now we start with the interviews. What's their potential? We assess how they work with different pH. We assess different types of nutrients. Can it fixate nitrogen or not? This has already been mentioned this morning. Also regarding biocontrol, insecticide. But the important thing is to know if the microorganism is able to solubilize some nutrients. So maybe we have to compare it with other microorganisms. Fortunately, we have characterized more than 120 microorganisms, and this enables us to understand the level our candidate is in. Does this candidate work better or worse than other candidates that are out there in the market? On the slide, we can see that yellow image, that's yellow. But if we compare it with feldspate, potassium feldspate, there are just a few microorganisms that are able to solubilize that compound. This way we can compare and understand how they work. The same can be done with microbial compounds. They can generate volatile compounds, enzymes, and we also have to characterize them. Enzymatic activity. This is the most important thing for us. When we talk about soil microbiology, we are aware of the fact of all the nutrients that are on the soil. This is a key factor. When we want to isolate something, we have to take everything into account. Okay, now we have a microorganism. Do we know what it can do? But we want to multiply it to be able to sell it. What if we are not able to multiply it and to sell it in the future? Everything is going to be useless. So we have to assess the multiplication capacity. So now we have selected a candidate. We have a pure bacteria. And that's where we start working. What's the next phase? The formula. It can be solid, liquid. It can be obtained via different techniques. But from now on, we're going to start working with a product that we want to sell. The feasibility. If that final product keeps what or maintains what we thought the product was going to do. Angela mentioned that in their formula potentiates an effect, and they have improved the result. Everything has to be tested in different places in order to adjust and to verify if what we thought the microorganism did in the lab can be done in the field with different variables: pH, temperature, solar radiation, humidity, in very abstract conditions. And the most important thing for me, dosage, moment, and the application time. And also mode of action. The secret is in the mode of action, as Jose mentioned this morning. We have to know why it doesn't work. We need to know the mode of action, the routes it activates, the ones it doesn't. We have to sell a product that has to be valid at least for 12 months. We need to understand the stability. It has to be stable at least for 12 months according to the legislation. We have a product that works, that improves or keeps the features or the characteristics. Now we move to the stage where we manage the contract with our candidate. Now we're going to register our product. We have to assess everything that is stated by the law, quality, routes. We can go a step forward. We can measure stress resistance. We can see what happens on the soil. Or what happens we at photosynthesis level. And we also have to take into account nutraceutical properties. We are aware that if we use these types of products, we can improve the quality of the final product, polyphenols, nutrients, etc. As Jose mentioned, improving color, increasing the amount of vitamins, and it's going to be better for the final consumer. And this is thanks to objectives that can be quantified, that can be compared and replicated, and that can be improved. And that's how we get a commercial product that has been registered for a specific use. So we have selected the candidate, and this is the moment to sign the contract. We're going to hire this guy. Now we have negotiated the contract, and he's going to join our group. I'm going to finish. Maria, don't say anything. As a conclusion, let's think about this process I mentioned at the beginning, as the headhunter said. We have to identify what we want. We see what plot is available or where we can get the microorganisms from. We isolate them. We eliminate pathogens. We make the pre-selection, in vitro tests. We select one. We negotiate the contract. We sign the contract and he joins the company. And then afterwards, there's the follow-up. It is really, the follow-up process, the monitoring process is really important because you're going to mix them. We're going to use them in different situations, and that feedback you give us help us learning more about the product. That would be the monitoring or the later follow-up to see how that worker joins the company and how he relates with his workmates. So we have to be aware of the complexity of the process. I think we are all aware of that. The investment and that it implies, and the amount of time it implies, at least 3 years, 3 to 5 years. The importance of knowing why it acts, how it acts, what can be expected, what cannot be expected, incompatibilities, in order to have a registered product. We cannot sell a product if it's not registered. And a user manual to know how to use it and how it works. And finally, I'm sure you are one saying, this is great, this is wonderful, but what's the use of it? I'm going to show 2 questions that I found on LinkedIn a few days ago. One was a colleague who said we have a very high levels of non-assimilable potassium. Can a microorganism solubilize potassium? And can trichoderma in a soil with pH 8 soil give me a good result against Fusarium in lettuce? I wasn't able to give an answer to those questions. Why? Fusarium in lettuce, you know how resistant it is, was the strain. I would ask the manufacturer, have you assessed if your trichoderma can control this strain? And what about pH 8? I have no idea. So we always have to take strains into account, the importance of strains. Each trichoderma strain is going to act in a different way, and it is essential to know it, because trichoderma can act generating antibiotic substances. It can activate the plant defense systems. So you really have to know your strain. And from then on, you can respond. And the easiest way would be to say, yes, my product can do that. And that's the aim of this conference, to try to give you a different point of view and for us to be more critical when using these types of products and to know how to manage them better. Thank you very much to all of you. And thank you for being here with us.
I didn't have to call your attention. Very good. Thank you very much. We're now going to continue talking about the challenges of draft. We're going to listen to Khalid Akdi from Tricodex, Fertiberia Group, challenging draft, key microbiome in the adaptation to water stress. Khalid, you have the floor. Can you please show my presentation? Thank you. In this world that will hunger for solutions and thirsty for solutions, draft is becoming a great threat, and where the revelation of the century is the microbiome. This microbiome that is a treasure, that is it's in the environment, associated with all ways of life or forms of life. This microbiome that is presented as the savior, our savior, vis-a-vis the stress caused by draft, is a new paradigm. But let's not get mistaken. We need a holistic solution with room for everything, for ideal agricultural practices, organochemical fertilization that is balanced, efficiently from a biological point of view, taking into account new knowledge. Well, after this poetic introduction, I'm going to continue with my presentation. The climate models show that the future is going to be complicated in terms of drought and the intensity of them. It's going to get gradually complicated, more and more complicated. We're seeing what has happened with the increase of degrees in the last 60 years. This has led to the loss of productive capacity representing 7 years. This is all very interesting, and many researchers have focused on this, us that we work with microbiome, to reveal the impact on plants. But what is going on with the microbiome? What is the effect of drought on microbiome? There is very few information revealing this impact on microbiome. We know more about the plants, but very little about microbiome. And since microbiome is an organ that is extended in plants and humans, we need to delve into this. And above all, we need to have a look at it from the field, because 90% of the data we have comes from controlled situations. So this is a call to deepen when studying the impact of drought on microbiome in the field. With this approach, microbiome as a response to drought, have had a mobility, a plasticity that is very important throughout many, many years. And microbiomes have faced drought, and this has involved many mechanisms and modes of action that we can divide in three main categories: climatization, change, shift community, and evolution. These three categories, the first one, climatization, is not an adaptation, it's a phenotypical change of microorganisms composing a specific microbiome that makes these face better drought or the stress as a result of a drought. And this can go from hours, days, or to months, but this can be reverted. This is a gene expression as a physiological function. Then we have changes that are deeper at community level, microbiome community level, and this can take longer. This is a result of the dispersion of microorganisms that, especially those with more capacity to face the drought, to find microbiomes and introduce themselves in soils where there is a higher availability of humidity. That's a result as well of a natural selection, but also we have those that have moved in those communities due to a better tolerance to this stress. And finally, evolution, that needs a lot of time, even in millions of years, in decades and millions of years. This is a change that can be reverted or inverted. The traits of microbiomes can be reverted. This is very interesting. What we don't know is how dangerous the content of carbon in the soil can be. We've mentioned in the morning that the microbiome is part of the biochemical cycle, especially in relation to the presence of carbon and the nutritional mobility in the soil. When the plant suffers a lot during a drought, there are very few inputs, not a lot of production of carbon. And what this happens due to these survival mechanisms is adapt and adopt new mechanisms to use the carbon from the soil. And that is a very important danger, or that has to be taken into account. Regarding strategies within the microbial community to face drought and results of drought, we have tolerance, resistance, avoiding, to face, to put your head down and adapt, or run away. Regarding tolerance, microorganisms strengthen their wall cells, especially bacteria and fungi, and they produce more osmolytes, and they produce biofilm war strategies to associate and produce substances in large amounts like auxins. But also they have resistance ability. They transform like spores, or they create aggregates that allow them to find refuge in a capsule, and these forms of resistance take them back to their original state after a specific content. And then we have dispersion. Many of these microorganisms have tools to move. They move, and those that are more adapted are looking for more favorable conditions for themselves, and that's a way of avoiding this. Without this, what happens when there's stress or draft, or the results of a draft? We have responses by the plant, we have responses by the microbiome, and the response of the plant and microbiome have an impact on the plant and on the microbiome in turn, in a direct and indirect way. At the same time, we have a response and an effect, and one affects the other, and vice versa. It's so complex, this feedback between the effects of the plant and the effects on the microbiome, but also they also affect the response of the plant vis-a-vis drought and vis-a-vis microorganisms, and vice versa. This feedback is so complex that we need more studies to reveal this, and I would like to insist that the knowledge we have is thanks to studies carried out in controlled circumstances. Tricodex has been working for many years in applied biotechnology applied to agriculture, but everything has changed. When we talk about new paradigm, everything has changed. We cannot keep talking about development methodologies with biofertilizers as we've been doing the last 40, 50 years. We cannot continue talking nowadays with of a microorganism or consortium. That is old now. There are consortia that are capable of taking a whole country to a very hard situation. The same happens with football. When we talk about consortium of microorganisms or individual microorganisms, when we talk about microbiome, we're beyond that. We have to delve into the forms of producing those microbiome. We know that microorganisms can adapt, can change, can evolve, and there are many techniques and technologies related to the knowledge that we have from microbiome that have enabled us to adapt to these new technologies and to produce biofertilizers that have quality and persistence, and can colonize the rhizosphere. So this is what in Tricodex we have developed technologies that enable us to produce these microorganisms as if they were in a specific microbiome using very advanced techniques. We have technologies that enable us to produce a consortia in response to specific inductors or individuals that can integrate in the microbiome and have an influence this microbiome. These microorganisms are very old, but are the same ones that we've known, but with different ways of production. Here we present 3 products. One of them is Vibacter, designed specially to regenerate plant microbiome, especially when there's low biodiversity, to improve the assimilation of nutrients and improve water stress. Then we have another product which is a champion in the solubilization of phosphorus, but especially to mitigate salt stress, and then a fixator of nitrogen with innovative technologies that can be produced put on the soil and in the leaves. I'm going to present some aspects that are common and are determined by the conditions, the control conditions in a lab, like all of them. And they reveal the ability of this microbiome. We have the formation of biofilm, and we've proved that all these consortia can form this biofilm, this tissue on a specific substance or surface, sorry. And this is very important for adaptation to produce auxins in large amounts. We have to think at micro level in a community in the surface of the roots that can produce micrograms of auxins, and this can change a lot in the plant, not only induce the root mass, but also affect the production of auxins by the plant. These are microorganisms that can promote growth due to auxins and phytohormones. They can produce even more in 133%, and the relationship with the microorganism. And the most important thing is that they have the ability to promote or enhance the production of roots, of absorbing hairs, to promote or enhance growth. 2 minutes. This is different with Pedro Barton had more time. Mobilization of potassium. We talk a lot about solubilization, mobilization of potassium. It has capacity to soften phosphorus, to provide siderophores, produce siderophores. We need to see what kind of siderophores, catechomins, different types of siderophores. They have production to produce volatile substances. Plants can smell this substance and this microorganism and promote growth. We have carried out tests showing that this consortia can multiply the production of auxins in situation of stress, whether it's salt stress, water stress, or stress owing to high temperatures. It has the ability to do lots of things in vitro. But in the plant, well, in plants we have had different tests regarding water stress, where we assessed biomass, the number of flowers, the presence of isotopes, the carbon, nitrogen, 15 nitrogen and 14 carbon, and nutritional analysis, and the enzymatic activity related to the antioxidant effect. These studies have enabled us to see the interaction of the microorganisms with the plant. For example, the increase in the number of flowers when the plant is not under water stress. All of them do it, but some of them do it even more when the plant is stressed, especially phosphorus solubilizers. And we know why, because this phosphorus goes straight to create the DNA, the RNA, the ATP, the membranes. Also an increase of biomass, that's quite important. 15 nitrogen indicates that when the plant is under stress, doesn't need nitrogen, and that's normal. It's looking for protection more than nutrition. 14 carbon says that under stress, microorganisms opens up stones, and there is a metabolic activity that is important regarding carbon. And if we continue, there is nutritional analysis here with potassium, phosphorus, magnesium, calcium, boron, copper, zinc, iron under stress. You need to start wrapping up. Okay. At enzyme level, ROS, reactive oxygen species, we have the catalase, maize catalase. The activity is for guaiacol peroxidase, is the machinery that is used in the activity to face oxidative stress. That's what cleans the roots, make them go back to their initial origin or their initial status or situation. We have also carried out studies of water stress. The most important one were in citrus fruits or in different seasons. In 2021, 2022, regarding 2022, we see a reduction of production up to 58.4% with the use of a strategy based on these microorganisms. We managed to increase this gap to 54.53%. This is 40% reduction of water from one season to the other, and this enabled to have a benefit margin of 13,000 euros for these farmers. Finally, with underwater stress, we've seen higher growth, and up to 36%, where you can see it in these pictures. And there are many trials or tests that show the capacity of these microorganisms to face water stress. To finish off, we know the mechanisms with water stress and salt stress. Some of them are very common, there are very small differences between one kind of stress and the other one. With the use of these microorganisms, they contribute auxins, different substances that balance the hormonal balance, and there is higher control, higher conductance, and the plant goes back to its original status thanks to these microorganisms. We still have a long way to understand how these interactions take place in the differences in the lab and in the field, but in the field we've already proved it. Thank you very much. Thank you very much. Thank you to all the speakers in this second session today. I'd like to inform you of a change in the program. As you can see, that we had in mind a breakthrough, but since we're lagging behind and we want all the speakers to be able to share their papers with us, what we're going to do is we're going to continue and we're going to skip this break. Fruit, coffee, everything is ready outside. So if you want, you can go out, grab fruit or coffee and then come back. But we're now going to continue with our papers. We're going to move on to the next one. Our next speaker, María Jesús Zanon from Certis Belchin. The title of her paper is Strategies for an Adequate Soil Health and Sustainability. You have the floor, María Jesús.
Okay, but let's see, because everybody's standing up. Please. Okay. Okay, first of all, thanks to the microbiome organizers for inviting us. As you can see in the title of my presentation, at Certis Belchin, we talk about strategies, we talk about sustainability, and we talk about health. You can see that everything is going to be focused towards this first part of strategies. And we have also mentioned that what we talk about when we mention sustainability. Sustainability for us is something that can stay for the future. So we are creating strategies that can remain for the future. Having said that, this is the Green Deal slide. We focus on development. We have to be aligned with that 2030 agenda. So everything we do, all of our effort, is based on 3 pillars. The first, that's pesticides. If we have to try to reduce pesticides in 50%, we are making most investments in looking for biorational solutions. We have to increase our biorational portfolio. When we talk about biorational solutions, we always talk about phytosanitary products that are registered phytosanitary products. We always have to talk about strains. What are the strains that have been registered with our phytosanitary products? The second pillar, and that's what my presentation is going to be about, is new technologies, because we work on biostimulation. Why? Because we have to give tools to be able in the future, by 2030, to reduce inorganic fertilizers and improving nutrition. And all of these, and in order to complicate everything a little bit more, based on looking for products that can be useful and that can be used by organic farming and biodynamic farming. These are active matters that can be used in organic farming. Now we're going to talk about microorganisms. We have focused on the soil. The first point is phytosanitary products that have been registered. We can talk about fungicides. These are strains that have been developed. We can talk about the plantarum species of Trichoderma troviridae. We're not going to focus on this today because we're talking about the two best known microorganisms related to diseases, soil-borne diseases. So what we are discussing today is, if I've got a trichoderma, if I've got bacillus, what do I have to use? When do I have to use it? What's my problem? What's my pressure? What's my population? What's the type of soil? And that is developed by Thertis belchi, but this is just one leg. We're talking about fungicides. Now we get to nematicides. Can we control nematodes? We started with nematini. We have the PL11 strain, Purpucerium ilacinum. It will be registered for horticultural products, and we know which part can be controlled. Where do we place it? For the control of the egg masses. Purpelucilium colonizes egg masses and also juveniles. This is very important because at the beginning it's different. So we're going to lower populations and we're going to reduce the risk. So this part is related to registered phytosanitary products. Today I'm going to talk about new technologies of biostimulation. First, we're going to talk about HITA. This is high yield technology. If I'm going to work with a biostimulant, if the product works, we're going to have an increase of the production. But we are not interested in production, but on how we get the fixation of atmospheric nitrogen to the soil. The creation of consortia of bacteria consortium. We've got Binelandi and the strain is AS80. And this is created by a Clostridium pasterianum strain AS52. What do we offer? We're talking about Azotobacter and Clostridium. We're working with anaerobian bacteria and with an anaerobian bacteria. We'll see the benefits of that consortium. And finally, we'll see how we can fight or reduce abiotic stress, improving the biological activity. And in order to do that, we use new technology in yeasts. But we're not going to use the living organism. We are going to work with specific strains. We're going to work autolyzed yeast and fermentation extracts. These are fermented yeasts. This is kind of a cream, and then we follow a specific process. We put it together with the extracts of fermentation and we get a biostimulant product that's high quality. Then we move to the problem and how we solve it. Here we can see how we can check. If we visit the ministry website, this is the amount of nitrates. So you know how the presentation is going to work. At first, we started working on the problem of Mar Menor, the Menor Sea in the region of Murcia. The first one is a general image of where the problem of nitrates is located, but we want to see the concentration. That's what we see in the map on the right with the red dots. There are problems not just in Mar Menor, but also in the whole territory. Regarding HYTA, the famous microbial consortium, these are the main actions of each different bacteria. If we talk about Azotobacter vinelandi, AES80, if we compare it with Clostridium, the first action is available. Both of them are going to favor the fixation of atmospheric nitrogen to the soil, and then it can be assimilated by the plant. Then we have also identified that each of them can solubilize different nutrients, mainly potassium. And finally, there's something very interesting, and is the structuring biofilm. And we have seen that in phytosanitary products, we've got bacillus. When we talk about biofilm in the rhizosphere, together with the different pathogens, that doesn't have anything to do with that structuring biofilm. That is just related to AES80, to the surname Vinelandi from Azotobacter. This is an alginate. That's a jelly-fine substance that improves the substance of the soil, of the substrate, and it is related to a reduction of the conductivity. Always working with anaerobian bacteria. Now there's the anaerobian bacteria. We're going to improve the fermentation that the Azotobacter doesn't offer us. So taking everything into account, what I have selected are the average results of all experiences carried out for leaf crops in the area of Mar Menor. Graphs today in this forum are not important. What we see is an increase in the yield, but also quality is higher. And this is not just based on sampling, but on measurements. This is what has happened with nitrates in the area of Mar Menor. This is the final sampling. we have seen a reduction in the accumulation of nitrates. I have also stated the type of soil, but this is going to be related with to the next part of the presentation. This is something we're working on. The conclusion for this type of technology, it's easy. We have a bacterian consortium with an aerobian and an anaerobian bacteria that enables mobilizing nutrients, it fixates nitrogen, and then registration. Sorry, the use. Use for all crops. According to the type of soil, this has also been proved with or tested with all the products we work with. This is a collaboration with the soil team of the Technical University of Valencia, and we work hand in hand with Antonio Lidón. We got the yeast technology, one of the 3 products that's applied via irrigation. And we have 3 different types of soil: sand soil with a low percentage of organic matter, an optimum type of soil, and the 3rd type is the worst case scenario. We used a type of soil that hadn't been used for 7 years at their lab. So in this case, we added our product, Pushy Plus. This is a biostimulant that's applied with irrigation, and the premise is that we increase the availability of microbiota on the soil. This had to be proved on semi-controlled conditions with a single application, and these are the results. In that sandy soil, after the application of Pushy Plus and measuring the respiration of the soil, we increase the respiration activity. That means we are increasing the activity of microorganisms on the soil. That's measured with the release of CO2 and the consumption of oxygen. In the 2nd type of soil, and that optimum type of soil, can we finish, please? According to Professor García Izquierdo, if we have a soil that's optimum, we can use as many products as we want. But that's opposite to what happened with that complicated soil that hadn't been activated for 7 years. So when we used Pushy Plus, we started activating it. What did we activate? We activated all the resistance structures of the organisms that remained on the soil after 7 years. And that's the conclusion, is that microorganisms are activated after applicating this product. So the key word is strategies. We have Valcure and HYTA for the fixation of atmospheric nitrogen, and Pushy Plus as a biostimulant. And the strategies are very well defined. This is just part of the soil strategy that has been presented today, but we go on working on some tools. And that's all. Thank you for your attention.
Thank you very much. Our next speaker is Enrique Quesada from the University of Córdoba, and he's going to talk about entomopathogen fungi from protection to plant production. Good afternoon, everybody. First of all, I'd like to thank the organizers for inviting me into this third edition of Microbiome. And I've been listening to all the papers this morning, and my paper now is included in the context of protection of crops and control of pest control. I'm Enrique Quesada, doctor in the Department of Agronomy in the University of Córdoba, and I'm going to refer to a new research line that we've developed in the last 25 years that is important at an international and national level. First, I would like to give some context. Although we haven't got much time, you know that when the arrival of the 21st century, the EU gets into a second greening with the regulation of the use of pesticides and with royal decrees defining action plans at national level, and more recently with a new regulation revealing lack of conformity of the EU with what is happening to be more strict with the reduction of plaguicide active matter by 2030. This is a regulation that is being discussed now and that shows the need for new measures to control pests and bad herbs, and it looks like the EU is gradually drafting new regulations to facilitate registration, and we're now using non-chemical measures. And I would like to give some context. We've talked about biopesticides, but there are natural molecules produced by living beings, insects, microorganisms, but also the use of living agents, entomophage organisms for the biological control of pests. So the use of entomopathogen fungi is part of the control of pests with entomopathogenic fungi. And I'd like to start talking about protection, that we're talking from a point of view of protection. I'm going to present this fungi. Who are they? There are several groups of fungi. One is a very interesting group with higher mortality in natural populations of insects, Ascomycota, Entomophthalmycota. There's a limitation for their commercial development, and the most well-known that are on the screen in the hypocreal order. They can be cultivated in an artificial crop. We have here these main species that are marketed in the world in infections, both in a natural way and Acanthomyces lecandi, Metarhizium and Cyoplae with different principles. How do they act? This is how do they act within the microbial control of pests. These are the only ones that act with through contact. Most of them act in the digestive pathway. What they do is they reach the insect and entomopathogen fungi reach through the infection. They reach the cuticle or epicuticle of the insect where they normally germinate, create some structure, produce protease, physical strength. Here you see inlet points. It fights the defensive system of the insect. The insect and the strain cannot stop the infection. The infection evolves, and there's something that's interesting that once the insect is dead, the fungus use the corpse to grow outwards, creating conidium, and we can see the kind of figure where you see that the fungus is using in the body. We've now described different non-usual methods that are very interesting, that are really recent, that they talk about digestive pathway in mosquitoes. How do we integrate entomopathogenic fungi? We see that the digestive pathway is very complicated. Synanthropic insects that live with men or because of men, cockroaches and termites. Grasshoppers, fruit flies, geobiont and geophile insects, invasive species, and insects of interest, vet and medical interest. There is a world market of myco-insecticides registered in the world, and particularly in Spain. If there are no novelties today, the last project was by VAS. There are 4 myco-insecticides that use Boveria bassian. By the way, all of them have the same aim, to control insects. Where do these entomopathogenic fungi live? Well, they infected insects, and their main reservoir was the soil. In the 21st century, since 2006, we detected its interaction with plants, and they're in the whole plant as epiphytes and endophytes. In this sense, throughout the morning, it's been mentioned when we talk about microorganisms, the fungical strain is the most important thing. There can be a million strains in the world. We need to be cautious when we talk about fungical or fungi strain. It's very, very important. The fungical strain. This is the one that has to contain the insecticide activity, and it's one of the main criteria delimiting its activity and its environmental competence. I can have 10 strains of this kind, and not all of them can adapt to the Mediterranean agriculture if we have temperatures over 40 degrees in a greenhouse. So the strain is fundamental, and unfortunately, if the situation doesn't change, the cost of registration is very important, and the trend that we have now in the market is to globalize products. The companies that manage to register a product, they put it in the world market, and not always that is that successful, because there's a strain from the north of Canada that even might not be a very effective strain for Greece, Italy, Spain. Very careful in this table that I prepared myself. The world market is important, but there are a few strains that have been developed in the world market. How can we apply entomopathogenic fungi with different strategies using dissemination by arthropods, pulverization of plant substrate, leaves, stem, branches, application in the soil, application in with its new ecological functions. Regarding these are new lines for many companies regarding the arthropod use. There are scientific papers that talk about the distribution, the use of pollinizators. As I was saying, there are many papers, there's a lot of literature in this regard in the US against natural enemies for the dissemination, novel use of predatory mites for dissemination of fungal pathogen for insect biocontrol. What we create is sterilized male insects that, apart from fulfilling its function, it can develop other things. How to apply them? In with a spray. It can be sprayed in arthropods to fight the insects in that kill palm trees and wood. There are many papers indicating different applications of fungi on plant substrate. One of the best applications is application in the soil, where we cause less impact, like in the case of white worms, active geophiles or inactive geophiles. 80% of insects causing problems in our agricultural system spend some time on this in the soil, and there are many products to apply on the soil. In the case of our research group, we experienced during the control of fruit fly in Europe and in olive trees, there is the development not as easy as applying the fungi in directly, but something that is less frequent is to find something that is active in the pre-imaginal systems in the soil, where the larva develops. And here we see how we transfer to the sector a strain from the Metarhiza brunion strain that is being developed by copper to be registered to control olive trees fruit flies in the wide area trial, wide spectrum trial. In the second part of my paper, I'd like to talk about the ecological functions that are new for entomopathogenic fungi, because recently we discovered that this fungi can associate with the plant in the rhizosphere as endophytes, as epiphytes, and little by little, this can let us enter a territory that's very interesting. That is, the applications derived from these special relationships that fungi have with plants, not only when protecting, but also in the production of crops. Because nowadays we can do something as interesting as carrying out treatments of seeds for the systemic protection of the crop, treatments in the soil that also result in systemic protection or transitory colonization, competence in the rhizosphere, spray in the plant that can lead to competence in the rhizosphere with amplifying the control area for chewing insects and borers. There's a very interesting scenario for production, companies producing seeds for nurseries. We need to protect it systemically through the cycle of the crop. Description of the translaminar action for borers. They can reach the larvae causing the mine. The existence of a priming, the contact of the plant with the root of the plant with the fungus can stimulate different substances. In studies carried out by our group, changes in the ecology of the plant that can be very interesting in the in production of different substances. Something that we asked recently is its compatibility, which is very interesting with all the groups that it's been improved with all the groups of agents where we can work in production, even with fungicides. We do it with a herbicide, applying it to the olive tree at the moment where the application of the fungus is done, when it is prepared for harvest. And through this kind of relationships with plants, here we see the second part of our paper. The presence of the fungus can make the plant have a better response to other types of stress, abiotic, salt, or even promote its growth. It's been demonstrated in many studies how to control diseases with entomopathogenic fungi. In our case, in several cereal and wheat and sunflowers, we show different results. Better iron nutrition with calcareum and non-calcareum soils. The absorption surface is worth taking into account. We have to see the ferric reductase in plants of cucumber and melon, the increase in the length of branches. Also, we have enzymes related with the growth, and I believe that one of the takeaway messages, some books regarding protection of crops talk about the division of the pest control measures are divided depending on how the plant responds to a biotic stress. And I think that in the current scenario of the system, the political scenario, the regulatory scenario, we are going towards the future. Microorganisms have to help us respond to the challenges in the protection of crops and the production of crops. And there's something we're going to lose, which is the important actions or immediate actions that when we talk with our clients, we need to start saying this product has to be used for the control of this pest, but take into account that we have to enroot it, and you're going to have more benefits for the plant. And that's a fascinating world, and that's a world that all of you coming from the business world have to bear in mind. These products are not as quick. Something that insecticides didn't have is that the plant is going to have other benefits. And with this, I'd like to thank the organization for inviting me. I thank my research group, and thank you for your attention.
Thank you very much, Enrique, for keeping it short. Now we're moving to Agustín Murillo from Alltech with the presentation Living Microorganisms or Microbial Compounds. Good afternoon. First of all, I want to thank you all for being here in Microbiome. Those of you and those online, I want to congratulate the organizing committee for reinforcing the microbiome brand, for overcoming the expectations, and for linking microbiome to technological expertise, knowledge. And I'm really sorry because there are lots of people at All Tech. This is the second time I have the chance to give a presentation at microbiome, and I'm going to share my knowledge. This is not my knowledge, this is knowledge from the company. So let's start. I would like to start my presentation with a sentence from Victor Hugo that says, "More powerful than the mighty army is an idea whose time has come." I don't know that there's no doubt that the time of biotechnology, of microorganisms, of soil microbiome is here. This is one of the biggest tools we have to face all the challenges that are there. This is what Carlos Garcia was saying. Europe is looking at the soil. That's the priority, the living part of the soil. I want to thank all of the people who have been researching for many years, who have been talking about the importance of the soil, such as Carlos Garcia. A big round of applause, please. And to all those companies that have been investing in technology and who have believed in research. I think that's very important. And that's where now it's the moment when we need it the most. But let's go back for a second and let's sum up and analyze what we talk about in 2019 and in 2021. In 2019, we talked about the use of microorganisms, on the importance it had, on the fact that using beneficial microorganisms, we could benefit the fertility of the soil, that we could increase tolerance to drought. As it was previously mentioned, this year, this has been a very complicated year, a very dry year. We have seen differences in the development of crops where microorganisms have been applied and not, without any miracles, because it's been extremely dry. But we have seen how crops tolerated the lack of water. By 2021, we talk about soil microbiome as a key element for sustainability, on the importance of strains. We said that when we work with microorganisms and with the soil, our long-term goal has to be a suppressive soil. Let's bear in mind that in a suppressive soil, we are able to develop processes that are more sustainable, processes where we can reduce the carbon footprint and the production had better nutritional features. And that's something we're going to be dealing with this evening. And this is something that has to be mentioned. The market is requesting for more sustainable and healthier products. So that is what we need to get. And the problem in our sector is that we don't know how to highlight it. What's Alltech? It's a company that's more than 40 years old with a wide experience on microbial fermentation. It's located in more than 120 countries. And it has grown that much because this is a highly curious company. This is our chronogram a timeline that shows different dates. The company was created in 1980 by Pierce Lyons, an expert on fermentation processes. Started the company in 1994. The agriculture section opened in 2003. The biggest factory was opened in Brazil. In 2008, we started working with nutrigenomics. It was opened in Lexington, in Kentucky. This is important because we knew that there were substances which led to some behaviors, but with nutrigenomics, we get to know what happens inside the organism and why those processes happen. In 2019, the company says, we have Alltech ECO2. Why don't we use it for agriculture? To reduce the carbon footprint, to get more sustainable solutions. So let's help our customers measuring the carbon footprint. And in 2022, by the end of last year, we got to get married, so as to say, and we got to be part of the Alltech family. But let's talk about living microorganisms and microbial compounds. Let's see if we can solve the doubts. Why is the moment now? Because we're ready for a new step. We need to be ready to share technology, because if we had mentioned this in 2019, we just talk about microorganisms, but this was something completely new. But in the end, what do we need to multiply microorganisms? We need a traditional fermentation process. That's easy. How do we do it? With sugar, microbial inoculum, and I get a product. I need to have a minimum concentration of 10 to the power of 7 to get a product. But what if we do it in a different way? Here, you've talked about properties of the different microorganisms. It fixes nitrogen, it solubilizes phosphorus, potassium, it induces resistance. But what if we know why these processes happen? What substances these microorganisms produce for this to happen? This has also been mentioned today here. Hormones, enzymes, secondary metabolites. What if we're capable of creating a precision fermentation? What if in our fermentation process we can fix variables like temperature, pressure, time, carbon source? Do we always have to use the same one? Do we have to add other products in that fermentation process? Do we only have to use a single compound, or can I generate both the compound with a living organism and a microbial compound? Do I have to multiply 1, 2, or 3 different microbials with a different concentration of different compounds? That's where we are at today. Palazón already mentioned it. Enzyme activity on the soil. Why do we measure it? Because microorganisms produce enzymes. And what if in the fermentation process we produce enzymes that are known to be present in all processes, such as phosphatase, such as urease, such as beta-glucosidase? Do we only have to have a single product, or can we use more than one product in a single compound? I also want to share with you a paper published in Agronomy that explores the bacteria exometabolome related to plant biostimulants. This was carried out by researchers of Spain. One of them is Carlos, who's really famous in soil microbiome at a world level. In Spain, we pay more attention to people from outside and not from inside the country. And what they did was to work with 3 microorganisms. They multiply them and they analyze and try to identify all the secondary metabolites. At the end, they identified 10,000 secondary metabolites, and 442 out of them are related to biostimulation, and 14 out of them are related with the production of indolyacetic acid. I could name all of them, but I'm sure I'm not able to pronounce all of them. But when I talk about cytokinins, these are phytohormones, these are not part of these microbial compounds. And this is what we have to work on. This is not a matter of microbial compounds. When we multiply the microorganisms, we can get many other advantages. And our CEO or founder said, when you have a why, you find the how. And look at the world in a different way, because we have to make a difference. And this is something that he always stressed. At Alltech, our tribute range is synthesized via precision fermentation. And please allow me to finish with another quote by Kevin O'Leary that says, nobody has a monopoly on good ideas. No one. We all have a good idea. Now it is the moment for all of us to stop, to reflect, to share, to collaborate, and to go on developing new ideas, to use biotechnology and microorganisms to face all the challenges that will come in the future, and to know how to use it well. And that's all. I finish with the same sentence. Please, let's dream the future with our feet on the ground.
Thank you, Agustin, and thank you for sticking to your time. Now we're going to continue with our next speaker. We have 2 papers before lunch. I hope you can wait. Now we're going to give the floor to Pedro Fernandez, agricultural engineer. He's going to talk about organic and exogen carbon, an arithmetic issue. Thank you, all of you here and at home, for devoting part of your time to these sessions. I hope that you learned something from today. In all of today and tomorrow, we're going to talk about microorganisms. And the organization understood that everything is basic, but organic carbon was essential to be addressed. So let's start. This paper is not. I like to talk about 3 words: sustainable, resilient, and green. These are 3 main concepts. Our public administrators have destroyed these 3 words, have eliminated the deep meaning, and this sector and us are the ones providing content. So I'm not going to mention these 3 words. The first premise: the soil is not spillage, a place where we spill things. We talked about dosage, carbon. These are keywords. It's not a dumping site. Not everything is optimal for agricultural use. What exogenous organic carbon is good and which one is bad? Am I a priest? Have you seen me dressing as a priest? Well, I'm not going to say what is good and what is bad. What is regulated is inadequate for my possibilities. Am I a judge? Am I dressed like a judge? No. So I'm not going to be answering these questions. Are all our soils in need of the same thing? No. We already have the Ministry for Equality. Soils mean something else. So, with these premises, what is important when deciding the introduction of organic carbon in the soil? We need an amendment because an amendment is not only carbon, and it's very difficult to deaggregate what parts provides with the different elements. Origin of the carbon. It's important to know the source of the material, the process of obtention. It's going to give us an idea of the quality of the product, the content in pollutants, salinity, heavy metals, pesticide residue, human pathogen microbiology, quality of organic carbon, proportions of humic and fulvic acids, labile and organic carbon, microbiological composition, quality, heterogeneity, functionality. This is what I pretend to achieve, but reality is much easier than this. This is the structure of this amendment, and also the content and proportion of nutrients so that they are similar to extractions, because we cannot only remember the datum of extractions but the nutrient provision. When we talk about the source or the origin, that is that organic carbon? Yes, that's organic carbon. So we don't need to say much that most of these pictures show a different carbon. The addition of exogenous carbon is going to be a source of nutrients for plants in different proportions, stimulation of growth, active principles of growth, enzymes, hormones that are going to favor plant growth, control of pests and diseases, because the application of organic measures is going to promote super soil. What we hope is that is with the correct doses. Improvement of the structure of the soil. Nobody works on this because it's not profitable anymore. Measuring filtrations, so apparent density infiltrations and retention curves of humidity. This is very important. Some soils provide a lot of information about these properties that were forgotten. Degradation of phytosanitary in polluted and degraded soils. Organic carbon is going to help me. If we talk about three concepts: fertility, health, and suppressiveness. With these three terms, there is a common element, carbon. And this is going to improve physical, chemical, and biological properties. But we cannot have to see it as independent things. We need to see them as interrelated elements. We have to provide content to these three wonderful words. Carbon from the sugar industry. The organization has uploaded a video of me dissolving 2 sachets of brown sugar in distilled water. We sent it to the lab and we analyzed it. What do we have? Organic matter expressed on dry matter, where all the substances are folivic acids. What can we read in literature regarding the application of this? That this is a product that increases the density of the soil. The soil is more impermeable, more waterproof. What are we finding? That it affects negatively to many properties, physical, biological, chemical, and even to production. What do I have to do? Sugar for the sugar industry. Carbon for biosolarization strategies. We have different tools, and if we use biosolarization tools in a good way, they're very powerful. This is the scheme that Carlos mentioned this morning regarding biosolarization strategies under a plastic biofilm. This is a soil, black soil with low redox, zero oxygen. The the picture on your right would be the presence of that shows that the process has finished. What is the information I'm going to take home? When I'm talking about cold soils, we need to reach anaerobiosis, we need 4 milligrams of carbon per gram of soil. Situation of Cartagena in October. When the temperature is high, Campo de Cartagena in August, you need at least 1 milligram of carbon per gram of soil. This would be equivalent to 2.5 kilograms per meter. This graph shows why some farmers say that biosolarization works and some say that it doesn't. Anaerobiosis phase with 2 amendments, over a month. Other amendments don't reach 15 days. How long of anoxia? How much time of anoxia? 15 days for some, 30 days for another. This could be one of the characteristics that say whether this has been correctly done. This is a cycle of 24-hour cycle. We see some amendments that during the day reach some levels and the other ones are higher. Here, biological properties, enzymes. We have an organic amendment regarding control, different profiles in depth. The ones at the top are the ones showing more differences regarding control, emission of CO2, more microorganisms, so more CO2. Energy, ATP. The first profile is the one with higher concentration of that carbon. When you apply an organic amendment to the soil, what you expect is that apparent density is going to be reduced. Statistically, especially in the first profile, this happens statistically. The other thing is really strange. This is an infiltration curve. The amendment enables it to infiltrate much more, and this affects the properties. Volumetric content of water in the soil depending different suction pressures. We see the different figures on the screen. The same soil with amendment and without amendment can retain higher amount of water, and under stress or water deficiency is something worth taking into account. Human pathogens. This is what the Royal Decree establishes as of 31st December. Any residue applied on the field has to be with no salmonella and E. coli. If you apply mud, you have to report it. We haven't really managed this very well. Or at least agriculture engineers. It's been carried out by someone that doesn't really understand this. You can apply all the E. coli and salmonella you want if it's mud, but you have restrictions with depending on the material. Human pathogens in an exogenous carbon. If these pathogens reach the field, it's very hard for operators working with this polluted soils. In this graph, I want to say that when organic carbon is applied, the rate of decomposition of phytosanitaries are increased. That it means that if I'm using conventional practices and I want to turn to organic practices, I'd have to adapt to the real context that I have of phytosanitary. In tests with fungicides and insecticides, results are promising. Salinity and heavy metals. This is compost. How can in 2023 compost contain 11% of sodium? How can this be possible? This means that out of 10 kilos of dry compost, more than a kilo is salt. And some people are surprised when they see the analysis. How can you find in commercial compost 21 active substances in the compost? Heavy metals. This royal decree from New Year's Eve determines this with different classifications A, B, and C. Why is carbon an arithmetic issue? Because it depends the number of additions or subtractions that each amendment contributes. Because my decision to buy needs to be based on evidence and not in what you think. And so what now? What? Probably there has probably not been a time with more aggression for the primary sector. All of us here, without agriculture and farmers, this would be a circus. Because we're not talking about forests, we're talking about agricultural soil. So there are systematic aggressions on all the fronts. The Palma volcano, the COVID pandemic, epidemiologists, doctors, when they talk about contamination of agriculture, we are not there and we're not even expected to arrive. And I don't understand why. Because this conference, if it stays here, it will still continue to be the same thing that we've done in the past. If we are not capable to leak this outside, to convey what happens here to society, we'll continue to organize symposiums and we won't have enough farmers. This is the language, the manipulation of language, ecological flow. What they're not telling us is that they don't want to send water here because they need to dilute the shit from the different plants. I'm an engineer. If we go down to Toledo, the content of nitrates in the Tagus river is 500 times more. And we keep silent when we see this ignorance. The ecological flow of the Tagus River in Andalusia, Europe, this bubble that is completely away from reality where they take decisions. There are no agricultural engineers there. There's nobody working on the field. Yeah, there are agricultural engineers that speak English. If you have not fluent in English, tough luck. Let's see if Google creates some translating system. Mar Menor, the small sea here in Murcia, everything that happens is the fault of agriculture. This is news from last year. Intensive agriculture is the cause of allergies in kids. What is intensive agriculture? Are we comparing it with extensive agriculture? This reaches society day after day. Are there any responses to these? To this? Nothing. There's nothing from our side. And this is the worst thing. At school, with children, they start to manipulate and indoctrinate with a message that is against the primary sector. What is going to happen in 10 years' time? When the best thing for the field in this time is this campaign by McDonald's, then we have a problem. We have two options. We can go to the bar. I love the bar. We can go to the bar and complain there at the bar. Or agroingenieros for agroengineers, for Mar Menor, for the small sea in Murcia. A group that was sick and tired of putting up with this situation and said, we're not going to take our guns out. We're going to take our knowledge out. Why? Because agronomy is a science very, very old, millions old, and it's still trendy, no matter what people say. Thank you very much.
Thank you very much, Pedro. Thank you. I'm sure there are many agricultural engineers who are willing to receive that message. And to close this session, the last speaker is Javier Vázquez from Compo Expert with a paper on biofertilization, living and sustainable nutrition. Good afternoon to all of you. It's a very bad time. We are all hungry. I don't know if I'm going to have a positive impact, but I'm sure it's going to have an impact and you're going to remember me. Let's hope so. My name is Javier Vázquez. I'm the technical responsible and R&D responsible of Compost Spain. Here we manage Spain, Portugal, and a great part of Africa. I'm going to show you practical cases of biofertilization with examples to show you what we've done in the past. It may be interesting for you. Since the beginning, we were at the avant-garde of nutrition. We have been innovating for almost 1 century in the world of nutrition and fertilization, with some specific cases that you all know, such as the brand CAFOS, the first soluble crystalline fertilizer created in 1927, and some other milestones that have become part of the sector, such as low-release fertilizers, different nitrification inhibitors, such as DMP, but there were many others in the past. And in the last 25 years, we have worked with microorganisms. One of the questions that came to my mind when I decided to make this presentation, I'm not very good at presentations. I'm a field guy. Is the application of biofertilization. Is it being implemented in the field? From Compo Expert, doing some calculations at an intensive level, we may be working in around 26,000 hectares. I don't know if that's too much. It's not a lot, but I think it is kind of relevant in order to see that things have been done and are being implemented. And we see that year after year, this is growing. So, biofertilization is being applied. And I'm going to comment the path that we followed from the point of view of microorganisms, incorporating them to biofertilizers and where we're heading towards. The first time we worked with microorganisms was in 2003 with our German colleagues, and it was focused on green areas. They started working on this topic, and this is one of my colleagues, Emilio Salas. They were products which created color differences, as you can see on the picture. Later on, we started assessing if there was a synergic action with a fertilizer, making a positive and negative control, and it seemed that there was a higher positive combination when used with fertilizer. So it could be really positive for fertilization purposes. From those green areas, we try to move to specific agriculture projects, such as the use of bacillus to microgranules to be applied around the roots. And this was proved at a very low dosage. We're talking about 20-40 kilos per hectare, but with a higher concentration of microorganisms. Then we saw we have biostimulants. We're working with them. They have a direct impact on production, such as the case of Eclonia maxima with vasfoliar kelp. And we combine that biostimulant that works very well in roots, and to increase the reaction in the fruit, we combined it with a bacillus. Here we have the negative control, positive control. This is vasfoliar kelp in 2 liters. The biostimulant effect with a higher effect in dry weight of roots and biomass. And Botanica are said. This is a combination of bacillus and this biostimulant that gives us higher results. So there's a synergy, as we can see. After these products, we moved to the use in fertigation that is essential for our area. Citrus tree, fruit trees, vegetables in all the Mediterranean area. There we introduced a pool of microorganisms combined with the nitrification inhibitor. In fact, this is one of the most popular products in our catalog. These products work very well. They're very easy to use. They're very easy to apply. And then we get results. And apart from this segment of fertilizers with microorganisms, there's another type of products that's also very important that have been mentioned before. These are prebiotics, organic compounds. There are different types depending on how organic matter is fractioned. This is a test with organic fertilization. Here, this is absolute control, positive control. This is mineral conventional fertilization. And then strategies with organic fertilizers. We also control our competitors, of course. And as you can see on the right, we see the strategy applying 1/3 of fertilizers, of usual fertilizers. T4 is 2/3 and T5 is 1/3, but in solid format. The important thing here is to see that we can have significant differences when applying an organic program. It has an effect on the crop, and we can make a difference at an organic level. Please do not pay attention just to the production results. For example, iron in the leaves, for example, foliar iron. Here we see interesting results for organic products. In the case of foliar nitrogen, we see that we're at the same rate, and in the case of phosphorus, maybe we had the biggest loss there. So the interesting thing is that according to the soil pH, we were able to make the soil more acidic in the organic program. And something important is that these fertilizers, almost 60% of sales are for conventional products. That means that it works. And when farmers see that it works, they use it. And some other impact regarding enzymes that have already been mentioned. With everything that we've seen with fertilizers and prebiotics, we wanted to go a step forward, deepening in biofertilization. nutrition of plants depends 90% on fertilizers and 10% on the soil. But that's not the case anymore. What we suggest now is to have a combination with a synergy between fertilizers and what microorganisms can give us with a soil activation. That's when we started working on a very important project to go beyond. As Pedro mentioned, we interviewed and we selected some microorganisms in different conditions. Here we see small plants growing on limescale stone or plants growing on the beach sand. Something has to be there because they're living in such hard conditions. We collaborated with Ideagro, with CIBAS, with CSIC, and it has worked very well. Here we selected some organisms and we looked for two main parameters: biostimulation and nutrition. That is generating more biomass, root, phenology, and to get to give that 20% extra to fertilizer. We did different tests from the dishes to mesocosmos to understand inputs and outputs. And this is the case of Lysobacter. Here we see a significant difference in the fresh weight. Here, a parameter that's going to be more and more important, and I have been told that is going to appear in some documents in the future to control what we introduce, how what we introduce in the soil affects respiration. That is something that we also assessed, and we saw how Lysobacter or how Strophomonas pavami had an important role on the soil respiration. When we incorporated the microorganism, the soil had a better respiration. It did not alter soil microorganisms, such as bacteria, mycorrhizae. And something that was very important for us was to get to that NPK balance that has been demanded at the region of Murcia. We take into account the input, the output, and we see what's left on the soil. And here we identified, for example, two strains with a high potential. For example, Lysobacter, which was able to generate more biomass and a higher nitrogen absorption, mainly with a higher extraction from the soil. In this case, regarding fixation, Pavani generated a very high rate of fixation. And the most interesting aspect is that this had a positive impact on organic carbon on the soil. So the balance of carbon on the soil is higher, so that's really important. These are all the field tests which were carried out with huge differences. This is a commercial standard, Azotobacter. This has been replicated with lettuce, onions, and broccoli. And we saw the evolution of nutrients on the soil. we had a positive impact in calcium, potassium, nitrogen, 8 to 12% higher levels, a positive balance in nutrients, the same for onions. And what we're going to launch after all this path is the Magna line of biofertilizers in granulate, granular, soluble, that works very well, and the pure concentrate with different types of product. And that's all. Thank you very much for your attention. Thank you very much, Javier. This is the first session. Thank you for being here, either on site or online. And now it's time to have a break, to have lunch. Lunch is across the street. We're going back at 4 o'clock. So have a nice meal.
Good afternoon. Please have a seat. We're going to start our afternoon session. Welcome again. We're going to start this afternoon session of the third edition of the Microbiome Symposium. We're going to welcome Elizabeth Sancho from BioEra, microbial characterization as a tool for the development of biofertilizers. Good afternoon. My idea is to share an interesting perspective in sustainable agriculture, and this is microbial characterization. In the last few years, there is a growing interest in the development of alternatives to conventional fertilization, and in this context is where biofertilizers come into place. But how can we find the maximum potential of a biofertilizer? This is where a microbial characterization is important. I'm part of the R&D team in BioEra, a company devoted to the production of biofertilizers with its own production of the microorganisms it works with. We have 4 production centers. 2 of them are greenhouses with an overall surface of 22,000 square meters, where we multiply mycorrhizae in an in vitro system. And then we have 2 other production centers where we have quality control facilities and processing and packaging facilities. The key points of this paper are microbial characterization, the different methods. I'm going to share some in vitro studies in the lab that we've carried out in the lab, the development of biofertilizers, and we'll go through the selection of strains, optimization of production of microorganisms that are going to be part of the biofertilizer, and we'll also talk about viability and effectiveness of the biofertilizer. And to finish off, I'll share some success stories on the use of bacteria in crops. Okay, what is microbial characterization? I think that I'm not the first one today to talk about this. I'll probably talk about things that you're already familiar with, but I'll try to convey the idea that I have, that you are able to understand what microbial characterization is in the most simple way. It's the study of morphological, biochemical, and genetic traits of a specific microorganism. Each trait has an effect on the plant, and we could divide them into plant growth promotion effect or protection vis-a-vis phytopathogens as an effect. There are bacteria that have the ability to solubilize phosphorus present in the soil, increasing the availability of phosphate in the plant. They do it through two mechanisms: the mineralization of organic phosphorus or solubilization of the inorganic phosphorus, producing organic acids. In the first case, it produces phytosis. Then we have bacteria like P. polymixa, fixating atmospheric nitrogen and producing and increasing the availability of nitrogen. Then we have strains like Pseudomonas fluorescens that produces substances that promote plant growth. The production of extracell enzymes by some bacteria promotes or enables the degradation of organic matter present in the soil and increases the macronutrients present for the plant. Bacillium subtilis can degrade gelatinase with the production of gelatinase. B. licheniformis produces amylases, and Bacillus megatherium produces proteases, degrading the cell wall. Then there are bacteria producing molecules that are siderophores, that we've already mentioned today, that have the ability to collect the iron present in the soil and presenting it to the plant. So this way they help this collection of iron. The bacteria is also benefited because when collecting this iron, it grows in detriment of other pathogens that are in the soil, limiting the growth of these pathogens and their survival. The production of biosurfactants and biofilm by some species of bacilli increase the ability to colonize in rhizosphere. The production of biosurfactants reduces the surface tension of water and enables the movement of PGPs. And the production of biofilm enables bacteria to colonize the roots of the plants. The bacilli species have the ability to create antimicrobial substances that inhibit some pathogens in plants. Here you have two pathogenic fungi, F. oxysporum and Alternaria species, that inhibit Alternaria species. And in the left, Santimonius campestris. In parallel to all these studies regarding characterization, we have to analyze compatibility to verify that our biofertilizers do not cause interactions with other products in the crops, like pesticides or other active substances. From our point of view, the key points in the development of a biofertilizer would be, first, to determine or detect a need in the market, like, for instance, to improve the collection of a specific nutrient by the plant. Then, based on the detection of these needs and using the results in the lab of microbial characterization, we would select the microorganism. Once microorganisms have been selected, we have to define different formulae and carry out stability studies to see that the formulae are stable and the life of the biofertilizers is long and that their components are compatible. In this point, we would carry out trials in crops to assess effectiveness and safety of the biofertilizer. And if this is correct, then we would move on to the escalation of microorganisms that are part of the biofertilizer in our pilot plant. And after this, we would just register and market the biofertilizer. The escalation of the pilot plant aims to optimize the production of microorganisms that are part of the biofertilizer. There we optimize the conditions of fermentation and downstream to ensure that it's an effective production. These are the fermentators where we do the escalation in our facilities. On the left, you have one of 5 liters, and the one on the right, 200 liters. After the escalation in the pilot plant, when we already have the optimization, well, we've already optimized microorganisms, the obtention of microorganism concentrate and of the biofertilizer, we move on to crop chamber tests to assess our biofertilizer. Here we can carry out different studies in different crops. And from then on, we can move on to studies on the field. We need to say that all the strains that are part of our biofertilizers have been documented and registered in the type of crop so that we can have them available in the case of commercial applications or future studies. And to conclude, 2 studies on the field regarding the application of PGP bacteria. The first one is the application of a consortium based on Bacillus megathelium in broccoli in the open field, 3 months, and we assess the effects on production, extraction of nutrients, and health of the plant. We conducted 2 sectors, and one was treated with a mixture of the consortium of 5 of 9 CFU per gram. And the treatment was after 15 and 45 days of the transplantation. So the final concentration was of 5 to the power of 12 CFU per hectare. After 3 months, after harvesting broccoli, we saw that the treated sector increased 3 tons per hectares in comparison with the control sample, and the pieces looked more healthy or healthier. When talking about extraction of nutrients at foliar level, there was an increase of nutrients for nitrogen in 16%, calcium 20%, magnesium 42%, and for manganese 41%. And the second study on the field was to verify the effectiveness of the PADQ program using different treatments based on consortia of microorganisms of bacilli in a complicated soil with salinity problems and root asphyxia. As I mentioned, we had different treatments here at different times and different mixtures with different doses, but all of them based on mixtures of different species of bacilli. And here, when we harvested 70 fruits, after 3, 4, and 6 months, there was an increase in the total weight of these 70 fruits of 10% after 3 months, 18% after 4 months, and 13% after 6 months. In the treated sector, if we compare it with the control one. And regarding the weight of fruits, we saw differences. And these 70 fruits were assessed, and we saw that in the treated sector, in the units over 200 grams were of 68%, 68, and the pieces above 250 grams were from 17 to 50 to 70. We saw here at foliar level the extraction of nutrients, and we saw it in global terms, and we saw there was an increase. 2 minutes left. There was an increase in the extraction of nutrients in the case of iron and manganese especially, and we also saw larger size and better color of the leaves. And that's it. This is my paper. I hope you have found it interesting, and I have conveyed my passion for this field, which is the microbial characterization, and I hope you are more aware of the power that microorganisms have to develop and evolve agriculture. Thank you very much, Elizabeth. Thank you so much for sticking to your time. And now we welcome Lorena Carro from the University of Salamanca, that is going to present desertic microorganisms vis-a-vis climate change effects on crops.
Good afternoon. I hope nobody's taking a nap because it's after lunch and it's kind of a difficult time. First of all, I want to thank the organizers for inviting me to share what we do at the lab and for allowing me to expand my vision. I've been working with microorganisms for 20 years in the lab, and it's fantastic to see that everything we've been working on for so many years is having a projection, and there's such a big conference at the national level with so many people interested. So it's a pleasure to be here today. I'm going to talk about the last project we've been working on, and it's looking in desert microorganisms. What are those that can help us fight the effects of climate change? These are graphs that I'm sure you are all familiar with. The huge problem for the future is to be able to feed the world population. But the problem is not just the population increase, but the fact that there's less and less available land due to different reasons. The increase of the population also increases urban areas, industrial areas, and also due to the degradation of soils. One of the big challenges is going to be to find solutions to those problems so that we can produce the amounts we need in the smallest area possible. In order to solve these problems, for years, fertilizers have been intensively used. This increased the production at a moment when it was necessary, but this also has some negative effects that we are living today. The production of these compounds also has a very high energetic cost and a high CO2 production, and this also affects or contributes to climate change. the situation is changing at a world level. This is more and more clear. This has been announced by scientists for years, but this is something that we have started living on our daily life. We have spoken about this general increase of temperatures and the amount of rainfall, sudden rainfall, and how this affects our daily activity. Spain, as the south of Europe, is especially affected by these types of conditions. There's a big part of Spain that belongs to this semi-arid area, and many areas are moving from semi-arid to arid areas. And now we have to start looking for solutions to see how we're going to face the situation, because it seems that this situation is unstoppable. So we have to see what happens when we use microorganisms from specific environments. Plants, as it has been mentioned today, depend a lot on environmental conditions. Plants cannot escape and run when their conditions are not the best ones. And they have an ability to adapt to those conditions. However, microorganisms have a higher evolutive rate. They adapt faster to those processes, and they help those plants to face those stress conditions. This is the concept of holobiont of the plant. We, as a group of associated livings at a molecular level, there's always an information exchange between organisms and the plant. What do we suggest? We suggest that microorganisms in deserts have been adapting to those stress conditions for many years. In the desert, microorganisms have a higher concentration of salt in the soil, a more limited availability of water, and a higher amount of radiation. And they have adapted to that for years. So for us, thinking that microorganisms related to these desert plants, that they have a more adequate features to the current circumstances, lead us to think that they can be adapted to other kinds of crops. So the question is, can bioinoculants help us stopping the processes that are going to be derived from the climate change? We think so. We took samples. We traveled to two of the driest deserts in the world. One of them is the desert of Atacama and the desert of Sahara, which is the biggest, warmest desert in the world. Both of them have very specific geographical circumstances, and they have very high stress conditions with huge changes in temperatures, with lots of stress factors. We started from our research when we tried to analyze the activity of the soils in the desert of Atacama. We analyzed actinobacterial biospheres, and we saw that despite of the fact that it is a desert, we found a high diversity of microorganisms that have adapted to those conditions. Then we designed our cultivation means to isolate some of them. For example, we had been worked with Micromonospora for years, that is found in the nodes of legume plants, and they promote growth. And we found them in the Atacama Desert. So we wanted to recover those microorganisms and assess the abilities that they had. In general, we also isolated microorganisms from the soil, from the rhizosphere, trying to recover that general diversity that can be adapted to those conditions. In some pieces of research, such as this one, we analyzed some genies. And here we find the diversity. They are not grouped in an area, but they are highly diverse. Here we also detected some bacteria living in the desert that are specifically associated to some plants in the desert, such as Dermacoccus. The next step we normally do at the lab is to select some of them and to sequence the genome. Having the full information of the genome enables us to study the features of those organisms. We can analyze their stress response, osmotic stress, cold shock, heat shock, detoxification from different types of metals, and general stress responses that are going to enable us to see what are the organisms that can better adapt to these conditions. We are also going to analyze all the genes that we've been talking about for the whole day, related to hormone production, indolyacetic acid production, the production of carotenoids, and we're going to analyze transporters of those molecules so that we see that they not only have the ability to produce them, but for them to get to the plant. Apart from seeing this, we are also going to confirm that apart from having the genetic material for the analysis, these materials are do work when they're tested in the lab. So we tested the growth promotion of the different matters, and we confirmed that they promoted growth, and we measured the intensity of growth. So it is easier to choose the best candidates. Here we are talking about the fact about tolerance tests, such as the growth capacity at different rates of salinity, over 18%. That's really high. And another stress, such as growth at different temperatures, resistance to water stress, and the ability to produce some compounds such as proline and betaine that are going to allow to the resistance of microorganisms. After many tests, we select some candidates. Here we selected some candidates from the desert to test them in Medicago sativa. And here we wanted to see what happened with salinity. Here we saw that there were microorganisms that were highly tolerant to salinity, but they were not the ones favoring the plant accepting that salinity. So it's really important to select specific candidates to see what of them are really functional. We saw that some of them were really functional when promoting growth, but when talking about salinity, was not induced in the plant, resistance to salinity. And we also saw the opposite case. Another test we normally carry out is to see that they have the ability to produce biofilms and that they colonize the root. The ability to colonize the root is essential for that microorganism to be active when applying it to our plant. Here you can also see these are all these dots we see around the roots, and some of them can even deform the roots. And this way, bacteria can enter into the tissue and colonize it internally. Apart from these general colonization capacities, many of the desert bacteria and actinobacteria are featured because they can produce different secondary metabolites. These are examples where we analyze the ability of these bacteria to inhibit the growth of phytopathogen fungi. And you can see the comparison between the fungi and the control. This is in vitro. What happens when this is done on the plant? One of the bacteria, Prima, we see how it controls the development. The first one is controlling, the second one, the bacteria promoting growth. You can see it in the graph. There's a significant difference. When we apply the fungus, all the plants are dead, but in the inoculation, they all survive. They promote growth. And we get that development level that we have in the promotion of the bacteria. That's why we talk about biofertilization, biocontrol, but many of these organisms have different abilities and they're going to act in different ways with plants and on their development. I would like to tell you a little bit about this project that we're working on. Now we have decided to find for a specific plant that could be found both in Atacama and Sahara Desert and in Castilla León. I come from Salamanca, that's why we wanted to look for a local plant. So we're going to use astragalus plants, and we have gathered them from the 3 areas. So with metagenomics, we're going to identify what organisms are what specific functionalities are related in the desert areas that are not in warm areas. Once we identify those organisms, we're going to compare them with the different strains. At the moment, we've got 400. We're going to get to 1,000 and see if we have identified some of those isolates that are important to improve that resistance. If that's not the case, we're going to carry out specific isolations. We're going to identify specific isolates so that we can create consortia for specific consortia that give us all those functionalities that we find in the plants in the desert. First of all, to see if they are effective in astragalus plants when they are subject to stress conditions, and then if they can be applied to other crops. I'm done. And thanks to the fact that Lionel is also in the project, we're going to do a follow-up with a microscope with all of the wonderful microscopes he mentioned before. Some conclusions. Binoculars allow us to eliminate or to reduce the use of chemical fertilizers and to contribute to reduce the effect of climate change. That we need to identify core microorganisms. That microorganisms from the desert can help us stop the negative effects of climate change and to improve resistance to drought, high temperatures, and salinity. That strains, actinobacteria strains in desert have a high capacity to tolerate extreme conditions. And finally, that identifying specifically associated groups to desert plants can allow us to improve the resilience in the crops. I want to thank my group from the University of Salamanca I am part of, and thank you all for your attention.
Thank you. Thank you, Lorena. We're now going to welcome Santiago de la Serna, that is going to talk about the productive impact of Poconia and the biostimulant power and polyphacetic power. The multifaceted biostimulant power. Good afternoon, everybody. I'm Santiago. I'm in charge of the technical department of Atlantica Agricola. I'd like to thank you for your attention. I'm going to try to be pragmatic in my talk and to try to provide some new insights today. Where does the motivation for this talk come from? Well, comes together with the responsibility of conveying something without repeating ourselves from Atlantica Agricola, and in the talks, not repeating what have been said this morning and what will be said tomorrow. So we're going to talk a microorganism that is not in the panel, which is Poconia, in relationship with what some speakers were mentioning this morning. When you start to work with it and you become passionate about that, I am going to try to show you the passion when you discover this kind of microorganism. And some of these passions are sent by our clients. And here we start to explore and we start to exploit the maximum ability that you can extract in this, that we've already had maximum yield. We see infinity, lots of results with an impact in yields, with an impact in growth. This is quite has quite a bit of an impact. I'm sure that you all remember Filomena. There was a lot of snow a few years ago, and that was called Filomena, this snowstorm. And this was the surprise that we found they were covered in snow. And so we started stressing it as much as we could to see the result. Atlantica Agricola is a Spanish company based in Vienna, and has 40 years of experience and is present in South America, I think because of the language. We started there many years ago, and now it operates in 70 countries. We are now devoted to biostimulants, botanic extracts, and in microorganisms as well. These microorganisms are centered on the idea of living soil, Atlantis cell line. There are a few of them, but today we're going to focus on Pocomix, mixing to try to understand Poconia. Why living soil? Well, I don't think we need to explain this, but because one of the indicators of a soil, one of the quality indicators, the physical, chemical indicators are very good, but the biological one is the most important one. And we all try to attack it with the tools we have to win that biological compound in the quality of the soil. And what are we going to achieve with this? Another concept that is quite interesting, which is the suppressive soil. It's been mentioned by previous speakers. Sometimes I explain it. I can be okay, and my blood tests can be very good. But when my intestine flora is not good, my gut flora is not good, then I cannot do certain exercises. Well, this is the same thing. If everything is under control and I'm eating very well and have less affections, I'm healthier and have less pain. Less pain. The benefits of all microorganisms is quite clear. This is a summary of what we've seen. Everything in body was mentioned this. Improve nutritional improvement, water improvement, major collection of water, improves growth, the quality of the root space. It generates strong competence. We've talked about siderophores, how they can trap iron, etc. But what happens with Poconia? I'm going to try in this framework to say what benefits we have, so you can try to explore it because it's quite competitive. And this comes from the fact that we also produce trichoderma, and we have an important plant of microderma. And when you try to fight against this endophyte fungus, and you start competing against it in all facets, it's going to be most benefited than any other trichoderma. So this goes for a commercial product. I'm going to talk about in vitro and real experience, because I think it's important to do it on the field, because sometimes it doesn't meet what you do in the lab. This is the first thing. How it induces the growth. We are going to try to see where this growth comes from, to understand what's going on. If we know what the impact is, we can design a better product, and the aim is to recommend it and see what the potential is and when to apply it. It has a big impact. Sometimes I find it tricky because this is created by snow. When the snow is gone, and when we arrived, we found this is the same variety, the same operations, but an inoculated one, and the other one is non-inoculated. If you read a bit about Poconia, some people know it because of the dual capacity it may have when fighting other elements, but the potential it has and that you can find is the biostimulant factors. I'm going to go through them both and to see what impact they can have and how you can combine them to understand the tools you have to solve specific problems. We're going to talk about potential for colonization, release, induction. Interesting things. Poconia is an endophyte fungus that is going to live in symbiosis with the root. I like fungi a lot because they have very interesting benefits that they need to preserve in the best conditions possible the plant to be able to live together in symbiosis. And you see strange things because they understand what is happening at every moment in time, and sometimes they do things when they're in being cultivated. They contribute something that is missing in a different environment. They select what nutrients they prefer at each moment in time. These are curious things that sometimes you don't understand why, but they happen because of symbiosis. Another thing that this fungus has is that it doesn't only colonize the roots, also the parts under the stem were colonized. What does this mean? That apart from colonizing the absorbing part of the root, nutrients come in and benefits come in through other structures, and this means exploring more soil than the ones involved in the cultivation. So here you see that you can explore roots, but on the right you can see in the image the pseudo stem, what you see under the soil or under the land, and that's where you start understanding certain things. You can hardly see it here, but this is the type of trial with Poconia. I'm going to try to comment this. There are some trials that we've seen with our colleagues from Agro to see what releases phosphorus and what releases potassium. Poconia releases a lot of phosphorus. Between the phosphorus strategies, it releases a lot and very quickly. Compared with trichoderma, it is also released because they release a lot of potassium, but a bit more slowly. There are two main elements which are limiting and can interact. And this it's interesting that this microorganism, even in a consortium, can be designed for this aim. But does this explain these differences in yield? Well, in this side here, we can see how to regenerate and the different places where you can have problems. It's also explained owing to an amount that can induce growth enhancers. What amount? Well, in the strains that can induce trichoderma, there can be twice as much. It releases more phosphorus with the same levels of potassium. It colonizes with other things the pseudostem, and here we see that it promotes growth. So it's very good. Here we see some effects with microscope to see how it colonizes some parts where the bacteria cannot reach. And we also see, creating side absorption hair. And this implies this buster with the same control that is quite stimulated. We have to see the pressure with an application on this microorganism. This is architecture and the stimulant effect. Interesting things. It activates with different pathways the compounds related to flowering or florification. What does this mean? This has a big impact, and this translates in cucurbits, for instance. You can see it. It's translated in the potential of female flowers that you have over the control sample. And if you have good nutrition, that's a potential that you can make good use of. It's curious because we repeated it in 6 blocks in 4 varieties and 2 localizations or locations, and it repeated 30%, 35% of the potential, or 30% melon, watermelon, or pumpkin. Something important as well that I use every now and again is the production of siderophores that we've mentioned at some point today. Poconia has an important effect on siderophores. I've used it in complicated areas, in calcarean areas where it's difficult to manage iron, to reduce the contributions of iron and to go back to recovering this iron that is stored there. And the change is quite firm regarding the change in color as well. What other things are interesting here? Now that we know this, it's very interesting to see how they're acting to generate interactions that are competitive. And these competitive interactions is an interesting concept. Why do we have to create consortia? Because with a consortium, you can generate different groups of some symbiotic microorganisms that are not competing between themselves to generate benefit. Sometimes things happen that are quite interesting when with a biostimulant, when we measure the weight of the root. Sometimes you don't have more weight of root, but you have further growth. Why? Because the rest of microorganisms that are injecting these nutrients can explore a larger land than in a different situation. It's interesting because if there are not many restrictions, we have these things that you need to understand. Why is it growing more if the root is smaller? So what can we do with this? How can we use these tools in this context? I'm an agricultural engineer and I'm more in the technical side, and our aim is to understand how something works, to design a tool and have it positioned in the market. Sometimes we have things, but we don't know when to use it or when to explore microorganisms or optimize microorganisms. This is a type of association that we carry out. We like to work with some symbiotic fungi because they're interrelated with the root, and they can think for the benefit of the plant. And what results do we have? Poconia makes us use it as a starting engine. With the production, with growth enhancers in the production of phosphorus, contribution of iron. It comes in very handy because if we use it as starting engine and we want to produce flour, we're going to potentiate this overexploitation, and this is money for the farmer at the end of the day. What else? Key resources that we worry about: iron, potassium, and phosphorus. How can we compensate these cations so that there is no lack of balance between them? Renovation of secondary architecture, even after some flooding, and how this synergy of different ways to collect mycorrhizic fungi and endophytes. Our line of living soil is out here. We have a stand, Poconia, featuring Poconia, Trichomix, Pocomix, Nitromix, and then Mycomix to the maximization of nutrients. We're outside. You're all welcome and invited. I'm very thankful for your attention. If you have any questions, I'll be happy to answer. Thank you. Thank you, Santiago. Now we're going to welcome Antonio Vallejo from the Technical University of Madrid, that is going to talk about technology of fertilizers to reduce emissions, inhibitors of nitrification and urease. Welcome, Antonio.
Good afternoon. First of all, I want to thank the organizers, Microbiome, Professor Carlos Garcia, for having invited me to be here at this conference, which is very interesting. My name is Antonio Vallejo. I'm a professor at the Technical University of Madrid at the Faculty of Agricultural Sciences, and I have been working for 40 years in agricultural sciences. I work in the lab, but also in the field. I try to represent what happens on a daily basis through different agricultural practices. You have to point out that broccoli, but not that much. Please, we have lost the presentation. Okay, that's it. In the previous presentations, they have spoken about intensive production in agriculture, the sustainable intensification, and that has to do with reducing the environmental impact of agriculture. A great part of environmental impact comes from nitrogen, what we call reactive nitrogen. Nitrogen that is not used by plants, by microorganisms, and stays on the soil. And this is one of the responsible elements, and that's nitrous oxide. Also nitric oxide, which is a reactive gas which generates tropospheric oxygen, also ammonia and nitrate. This is what I have been working on for the last years, and the first part is going to focus on nitrous oxide. As you know, nitrous oxide is a greenhouse effect gas. It has a negative effect because it's only reduced in the stratosphere, and it reduces stratospheric ozone. It is the main greenhouse gas associated to crops, and the cultivated soils produce 3.3 teragrams of nitrous oxide per year. And why is this gas produced? I think I have been invited to the talk because of this. I'm not a microbiologist myself, but it has to do with the production mechanisms of N2O and NO, nitrous oxide and nitric oxide. These are processes that I'm sure you've talked about. Nitrification, that's passing from ammonium. There's a first step where hydroxylamine is created. Microorganisms, nitrificating organisms, do not make the most of all the nitrogen and release it as nitrous oxide. In nitrification, NO2 is always produced. In the second step, there's also a loss that produces nitric oxide until it is a nitrate. And in the nitrate, that's the iron that plants take that is necessary for the crop. Plants compete with microorganisms. And nitrate sometimes is used by microorganisms not just to create its structures, but they also use it to get oxygen from the nitrate and use it as an oxygen molecule. And that happens when I have a soil with not a lot of oxygen. When, for example, when it's wet, when it rains, when I have just irrigated. Because microorganisms need oxygen, and that's why they generate this nitric oxide. And this substance can also become nitrous oxide can also become nitric oxide depending on the crop. So nitrification is highly favored by conditions, for example, a high temperature, pH over 6. In Galicia, there are high amounts of ammonium on the soil. The water-filled pore space and we need it, and also the amount of ammonium. Ammonium is what these organisms feed from. What has happened that in the last decades we have been feeding ammonium, urea, and the nitrifying activity has increased a lot compared to a natural soil. If we see a graph on how gases are released or emitted, Here we see that NO is emitted when it's released when the soil is dry, N2O when it is wet, and N2 in rice fields, for example. So, what do we do to mitigate these losses in agriculture? Because we want agriculture to be sustainable. We have seen many agricultural practices that mitigate this situation, reduce the amount of fertilizer and adapt it to the plant needs. If the amount of nitrogen is low, the plant is not going to fight with the microorganism. Then fertigation, and we also have this third point, that's the use of inhibitors of nitrification that can reduce from 40 to 70%. This is a path that has been found by the chemical fertilizers industry, and these fertilizers are called stabilized fertilizers. And then we have other practices such as the use of legume plants, water management, etc. But I'm going to focus on nitrification. What does a nitrification inhibitor does? The commercial ones are synthesis products, they are chemical products. What they do is against what has been said here today. They don't kill nitrifiers, but they cannot expand. They block this activity. They do so in bacteria, but there's another path through other organisms on the soil, archaea, but the abilities are very low. So the inhibitor blocks that first transformation. So when I use a fertilizer with ammonia, if I block it, ammonia is going to stay longer on the soil. It's going to release less gas. So we have these three inhibitors. MPP and nitropyrin, DCD, DMPP and nitropyrin. Many works have been carried out about nitrification inhibitors, and this is a meta-analysis carried out to see the reduction, analyzing all the papers that have been produced all around the world. When inhibitors are mixed with chemical fertilizers, it can be reduced at around 44%. We are talking about DCD and DMPP. In organic fertilizers, for example, purine, this is ammonia. This is ammonium, sorry. So inhibitors are also recommended in some countries, such as in Germany. And in this case, the percentage is around 35% of the reduction. These are pieces of research that we have carried out to study the mechanism, because I want to know what the mechanism is to be able to intervene. This is a test, a field test, sorry. Here you can see the white bars. This is N2O. And we can see what part comes from nitrification, what part comes from denitrification, and what part comes from the remaining nitrogen that's on the soil. In gray, we can see the part coming from the soil. Without inhibitor, we see that there are associated losses of NO2 after some days. With the inhibitor, we see that the losses go on. And below we see a summary. So we see that there are some peaks. So this is not constant. This is emitted for 3 or 4 days. These are high peaks. This is another result of NO. We do this with automatic cameras in the field that are constantly monitoring. On the left, we see nitric oxide. The red line is urea, and the blue one is urea with an inhibitor of nitrification. When I have the inhibitor, emissions are below 0. What does it mean? That it inhibits more than the soil without fertilizer. And it captures NO from the atmosphere and it consumes it. How much does the effect last for? From 4 to 6 weeks. It depends on temperature. And they have a problem. And it's that the effect finishes and the inhibitor stays on the soil for 3-4 months. But the inhibitor slowly degrades. And these are chemical compounds. They can be beneficial in case there's a heavy rainfall, but normally it is a problem for DCD. That 3-4 years ago, it was recommended in New Zealand. It went from the soil to the milk and it was forbidden because it doesn't degrade. It doesn't degrade. What are the effects of nitrification inhibitors in the leaching of nitrate? This is something I worked on some years ago. Sometimes I saw that leaching was reduced, sometimes it wasn't. Here we see there's a reduction of leaching, but it's not important. Why? Because when you have nitrates, if you have a heavy rainfall, no leaching is going to take place. More qualities of inhibitors. We've seen that sometimes it's like a second cover in bakery performance wheat. Regarding volatilization of ammonia, this doesn't have anything to do with microorganisms, but it has to do with enzymes. The volatilization of ammonia is in a balance with ammonium. It is released to the atmosphere. And to avoid losses, it's important for ammonium to touch the soil. If we talk about purines, it is important for it to touch the soil. Why? Because of the positive and negative reaction of the different compounds. So why should we use a urease inhibitor? Because urease is very good, it's like a quality criteria that microorganisms are working properly, etc. Urea transforms into ammonium through urease. This is a hydrolysis. Ammonia is created. This is a representation. On the left, we see a ball of urea on the soil, how the pH changes. It got up to 9. And if I use a urease inhibitor, hydrolysis is lower and the pH is not that high. Everything is transformed into ammonia. That's the same graph, seeing how the pH changes. That's why the urease inhibitor, in some countries such as in Germany, they have forbidden urea for this reason. These are examples of how this works in the field. I'm going to skip this. And I just want to talk about performance. Nitrification and urease inhibitors do not have an effect on performance, on the yield. The dosage has to be really adjusted. And finally, everything that I have explained, these are chemical compounds. What's the way? What do we want to work on? What we want to do now is to look for nitrification or urease inhibitors, but with a biological origin source. We're using exudates from roots such as Bracharia that, in order to defend themselves, they generate nitrification inhibitors. So we're trying to implement those genes and to try to find natural substances from extracts and from microorganisms that have this capability for biological agriculture. This is the final summary. There are many more things to say, but we have to take into account that denitrification or nitrification inhibitors have not been included into the law yet, but we're hoping for them to be in the future. This is our team. Thank you very much.
Thank you, Antonio, for sticking to the time you were allocated. Those of you who are leaving, please don't leave, because there is some kind of surprise now. So if you're about to leave, please stay a bit longer. I'm really happy to be here today. If you still want to hear more live music, I'll be at the Nelva Hotel this evening at 9 o'clock. Thank you, Jesus, so much. And now, after this musical moment, we're going to move on to the last paper before the fruit break. So please stay. We're going to welcome now Juan Manuel Lopez from Provelte, that is going to present agriculture microbiology as a solution beyond the concept of product. Welcome, Juan Manuel. You know, you have to point at the broccoli. I've never had someone singing right before me. Now you've raised your expectations. I'd like to congratulate the organizers consolidating this third edition of the Microbiome Symposium. I think that the main challenge ahead of us, especially for marketing people, is to sell this, because I've counted about 100 and something strains today. So how are we going to sell this now? It's going to be very interesting to see how that happens. We've moved very quickly from one technology, from MPK to nitrate, to hundreds of strains and microorganisms that do lots of things. And frankly, it's good news. But now we have to recommend it and be able to work with it. I work in Provelte. I've been working for 1 year in Provelte, and I'd like to introduce the company first of all. It's been 50 years in this sector, and in microbiology, it wasn't well known. And I promise that there is great knowledge in the company. Our role is to introduce it in the market and take it outside. But after 30 years of research, we have plots that are 17 years. For 17 years, we've been repeating the application of a consortium of microorganisms. 17 years carrying out analysis, and this is of enormous value. That's Provelte. It's here 10 kilometers away. It's from Murcia, local company, yes. And in Provelte, we do the whole process. We cover the whole process: isolation of the strain, fermentation, production, and packaging. So we have a very deep knowledge of what microorganism entails. And my aim today is to share with all of you the knowledge that Provelte has, and especially to reflect upon what all of this means. I think that everybody has introduced very well what this is. If we talk again about microbiome, well, it's something that we've already gone through, but I'm going to try to bring some reflections upon us. We need a clear objective. That's very important. What do you want to do? What do you want to improve? What is the aim with microorganisms? At the end of the day, well, this is good for everything. No, what do you want to achieve with this? The second thing is that if I'm going to introduce a microorganism in a microbiome, in an ecosystem, in which one and why and for what reason? What is the aim and what's the action that I wish to achieve? When we see so many microorganisms, so many strains, well, we think that this is definitely going to be interesting. We've talked about microbiomes. The plant in itself has its own ecosystem, the soil has its own. But here there is an interaction between both of them, and that is very interesting when you see that there is a long interaction with tests, locations of crops. You realize that something's going on there. And this is a challenge for engineers, for the technicians involved in agriculture, to try to make sense with this. We come from a concept that is I apply this, the variety does this, and now we've changed this dramatically. Now we say, no, don't remove this. If we put this here, it acts in this way, ISR, whatever. And we say, what are we going to do now? There are a lot of things to discover here, lots to discover yet. In Provelte, we always started from the deep knowledge of the strain, and we've stressed this a lot. Strain is the important thing. That strain that you've isolated, you've removed it, you know it very well. This is very interesting. In few time, we've adopted different technologies very quickly. All companies have microorganisms now. Here, in the snap of a finger, I've got it. I can assure you that after asking a lot with the research team at Provelte, which is a magnificent team, after 30 years, we still research the genetic characterization of each strain in Provelte, because we keep being surprised. And this is where our starting point, this knowledge, deep knowledge, or synergies. This is a case of a biofertilizer consortium of bacteria. They have synergies between them. We have to confirm this, that there are no antagonisms generated, that it activates and is this microbiological buffer. And what happens when I apply this on the soil? What happens then? There are many interactions there. How does it work? Does it work correctly? Do they respect each other? Can we reach the objective? Well, there are two responses here, rigor and time. Rigor and how you proceed. Do you act properly? Do you know it well? And the time you need to prove that this is right, you need time. This is not just something you do overnight. You need time. And as the more time we have, trials, long term, you still need more time. Microbiome is more than microbiota. We have fungi, we have bacteria and nematodes. Some of them generate metabolites, enzymes. We see that there is an impressive process going on the soil, and here we add another element in the system, or two or three or seven. We include them in the system and they start working, and they generate more metabolites, more enzymes, etc. These dynamics are quite complicated or complex, but from the companies we try to make them more standard. We try to get them to help us in all cases. Here there are four things that are quite interesting, and that starts with the internal knowledge we have and on the field. The first thing is the obtention process. I can ensure that my fermentation process and what is in the fermentation liquid, that is not being polluted. In Provelte, we sterilize the different fermenters every time. And the second thing is consistency. Is it consistent? Does it work? Because I'm going to use it here. I'm going to use it in Badajoz, in Italy, in the United States, in Mexico. It has to be consistent in this application. Persistence. How long does the product respond. My aim is that during the cycle, there is enough phosphorus and nitrogen for the production. Is that my aim? I'm sure that I have to apply a doses. What persistence does it have? Whether it applies fertilizers or not, we have to find it. Toxicity. How can I explain this? If I introduce this or microorganisms in the soil and a great amount, we have seen in many trials that the doses I apply, if I get too carried away, production does this. This is not in line with the dose I apply. There is a process in this buffer that starts telling you, yeah, this way you can do it, this way you can't. So this imbalance or lack of balance that is produced, well, sometimes we talk about consortium in a very light way, but we need to bear this in mind, that we have to see this. Please allow me to present the map of microorganisms in Provelte. We have 12 we have Azospirillum brasiliens, 2 of those, Pantio dispersa, and then 5 bacilli. Some of them are for biostimulation, some for fermentation. Some of them are, all of them have been characterized from a genetic point of view. We know how many metabolites they generate, the amount, which ones they are. All of them have been manufactured in Provelte, 10 kilometers away from here. And these strains are protected, they're not ours. Microorganisms don't belong to anybody. It takes many years to have a strain, to characterize it and protect it. Yes, but you tell us this sounds very good, but tell us something about a trial or a test that you've carried out. Well, I've brought this test here, but in the mid-long term, with a name that is quite clear. It was carried out in Albacete in a variety of almond tree that was supposed to have a production capacity based on a fertilization strategy. This is something that's interesting as well. The varieties express production and different potential depending on what you do, because with the microorganism, with the fertilization, we have talked about flowering, about, well, this may happen. So we have this variety from a yin in Albacete, and the aims were very clear: to prove the efficiency in nitrogen and phosphorus. Very simple. Fixate atmospheric nitrogen and fixate phosphorus in the soil. Very simple. But reducing 50% of organomineral conventional strategy. The aim was to reduce fertilization? No. It was to verify that your consortium can provide value in an environment where you're doing is reducing units of fertilizers to see where you can reach. In this case, it's 50%. In other cases, 80%, 30%, because there isn't like a rule. You have 2 minutes left. There is no rule, a clear rule. You say, no, mine is this, mine is that. No, no, no. We have to be careful with this. The second aim was to improve the quality of production, to see what was going on with production. No more, I'm not referring to more kilos. I'm talking about more quality, a balance in the production. And what happened with the availability of nutrients? Because there is a basic principle in Provelte. We know that the aim is to make nutrients available. Microorganisms are working there, and the crop takes in the nutrients. What happened? In 3 years plus 1, 4 years, in a variety, in 4 years, we managed to increase in 400 kilos per hectare the production. Is this a lot? Well, might be. The variety has expressed a higher possibility, and this is there for a reason. What have we done with the indicators? Macro and micro indicators. We've seen what happened, the dynamics. In this case, and this happens in this case, but it's also repeated. So what you find is a stabilization of nutrients that are available and soluble. It's as if they are working and they show it there. They are available. And what does the crop do? It feels comfortable. Oh, I'm available. So I'm feeding myself. And that's it. Thank you very much. Thank you very much.
Good evening. We're going to go on with the last part of presentations. We have 5 presentations, and as soon as they finish, please do not leave because there's more. There's a cocktail. We're going to have some drinks, and we're going to do some networking. We're going to start with the first presentation by Leo van Overbeek from Wageningen University, Harnessing Plant Resilience Against Disease and Pest-Causing Agents by Adaptation of the Microbiome. Gracias. Bienvenidos a todos. Soy Leo Overbeek, trabajo en la Universidad de Wageningen desde hace 30 años con microbioma desde un punto de vista científico, aunque colaboro mucho con los profesionales y de eso va mi charla. Es un proyecto que abarca no la parte científica, sino que se basa en cultivos que se producen en Países Bajos, especialmente en invernaderos, lechugas y tomates, principalmente. Esto es una colaboración público-privada. En primer lugar, está financiada por el Ministerio holandés de Agricultura, pero también por empresas de semillas y asociaciones de agricultura y horticultura. El principal objetivo de nuestro equipo no es hacer investigación del microbioma desde el punto de vista académico, sino desde el punto de vista práctico, desde el punto de vista de la empresa, porque hay un gran vacío en este aspecto. Y queremos saber qué puede la práctica aprender del conocimiento del microbioma y cuáles son las aplicaciones prácticas del microbioma. En primer lugar, un poquito de historia, porque empezamos este trabajo. Mis compañeros en los invernaderos trabajaban mucho tiempo en la lucha del TRIPS e intentaron controlarlos con endófitos. Aquí vemos las plantas y cómo se ven afectadas. Y luego me planteé, ¿qué podemos hacer con ello? ¿Qué sabemos sobre su forma de actuar? Hay distintas cepas de distintas especies. Entonces, ¿cómo interactúan? ¿Cuál es la ecología? Y por último, pero no menos importante, ¿cómo influyen a la resistencia de la planta? Es decir, ¿cuál es la protección de la planta contra los patógenos que se traducen en el metaboloma y el microbioma? De nuevo, los 4 agentes que vemos aquí en el recuadro, pero añadimos más de nuestra colección. En primer lugar, tricoderma, aislado de un bosque, y lo interesante es que también es tricoderma viride, es la misma especie que el que vemos en el recuadro, pero la actividad es distinta entre ellos. Un par de endófitos para trabajar contra TRIPS y nunca se han registrado porque están relacionados con los patógenos humanos y su registro es muy complicado. También Pseudomonas putida, os contaré más acerca de ella más adelante. Es un patógeno muy importante. Luego, Pseudomonas fluorescens C17, y otro es Bacillus mycoides y de nuevo viene de la patata. El último no es un endófito, pero somos los únicos en el mundo que hemos aislado esta especie y viene de un grupo completamente desconocido, así que no sabemos qué hacen. Así que estos resultados son fascinantes. Principalmente nos centramos en los tomates y en las lechugas. Tratamos el trips, pero también lo tratamos con de un hongo patógeno. Por otro lado, vemos el tratamiento biológico, que se trataron con las 10 cepas independientes en las plantas individuales y añadimos el patógeno y una combinación del patógeno y lo hicimos con todos los cultivos y además investigamos la colonización y el impacto en el crecimiento y en el sistema de la planta. Aquí vemos la situación general y toda la parte sistémica de la planta, comenzando por las raíces. Analizamos el microbioma, la parte de los hongos y las bacterias, el fusarium en el caso de los tomates y las lechugas, la endosfera. Nos centramos en la base del tronco, del tallo. Y luego en las hojas investigamos la metabolómica. Aquí vemos los resultados de la colonización y lo primero que vemos es que todas las cepas fueron capaces de colonizar las raíces del tomate y esto fue una sorpresa. Algunos también colonizaron el tallo, los tallos. En negrita vemos los que se utilizaron para los tomates y las lechugas y vemos que también colonizaron los tallos. Además, medimos los efectos, pero no encontramos efectos de los agentes biológicos, pero en el tomate y la lechuga también vimos los efectos de los agentes patógenos. Y en el caso del crisantemo, vimos el efecto de los trips en las hojas, en las flores. Esto es un poco complicado, pero a la izquierda vemos un tomate y aquí vemos la C de control. Estas son las plantas tratadas contra fusarium, pero cuando añadimos los agentes biológicos vimos un efecto muy distinto. Cada uno se comporta de una forma distinta en la planta. En la flecha verde vemos que estimula el crecimiento pero el rojo vemos que reduce el crecimiento de la planta. A la derecha vemos el caso de la lechuga y los resultados son aún más interesantes. Vemos una reducción del crecimiento. Así que vemos que si nos fijamos en las flechas verdes, vemos que el patógeno no reduce y esto aumenta la resistencia de la planta. Así que lo que está ocurriendo aquí en la lechuga, que no ocurre en el tomate, es que reduce el crecimiento por primera vez, pero las plantas son más resistentes. Y la flecha azul es Trichoderma viride que aporta una mayor resistencia. Ahora, si vemos el microbioma, analizamos la parte bacteriana y la parte fúngica. La parte superior izquierda vemos una clara separación de los distintos microbiomas de cada planta. Aquí vemos los tomates en naranja, que son totalmente distintos del crisantemo y de la lechuga. Tenemos parcelas tridimensionales. Los ejes no tienen dimensiones, por eso lo llamamos adimensional. Y cuantos más cerca están, más se parecen las comunidades entre sí. Aquí vemos en un color distinto los cultivos que fueron tratados contra el patógeno, y esto viene determinado por la presencia del patógeno. Está muy bien, pero si nos fijamos en los hongos, la situación es bastante distinta. De nuevo, en los tomates hay una clara separación, pero si nos fijamos, la lechuga está más difusa. De nuevo, cuando vemos la presencia o la ausencia del patógeno, vemos que en la parte inferior está el tomate y el patógeno está separado, y también la comunidad fúngica aparece separada de la presencia del patógeno. Si vemos los detalles de cada agente biológico que aplicamos, aquí hay muchísimos datos, los que se han recopilado. Esto es bidimensional. Vemos triángulos morados, que son el control. Los 5 controles están muy cerca los unos de los otros, lo que quiere decir que no hay grandes diferencias. Cuando utilizamos el agente biológico, vemos que está separado y vemos que la comunidad ha cambiado por la presencia del agente biológico. Y sin el agente biológico, vemos que esta diversidad aumenta enormemente rápidamente. En combinación con el agente biológico, vemos que vuelve a separarse. Esta es una imagen muy clara donde el tratamiento con el agente biológico tiene un efecto muy claro en la inmunidad. Y así es como hemos investigado todos estos datos. Todos estos datos los obtuvimos del metaboloma, del microbioma bacteriano y del microbioma fúngico. Lo hicimos para las 3 especies y lo hicimos según un escenario, que es como llamamos a esto. Voy a explicarlo. En primer lugar, comparamos las plantas control, es decir, sin tratamiento, con las plantas tratadas con agente biológico, y comparamos las plantas tratadas con un patógeno con las que estaban tratadas con un patógeno y agente biológico. Y entonces tenemos distintos escenarios. Uno, no ha ocurrido nada. El segundo, se añade el agente biológico y no. El escenario 2, ocurre algo, pero solo entre el control y el agente biológico. Lo contrario ocurre en el escenario 3, es decir, cuando el patógeno está presente, vemos que hay un cambio. Y por cambio quiero decir una diferencia estadística. El cuarto escenario es el más interesante, es en el que hay cambios en ambas situaciones, es decir, si tenemos un agente biológico hay cambios en la planta, y si utilizamos todas las combinaciones hay cambios en todos los casos. Este es un resumen de 3 años de investigación en una diapositiva. Parece complicado, pero en azul podemos ver el microbioma, y fun quiere decir hongos, bacterias, alfa y beta son los distintos tipos de diversidad alfa y diversidad beta, pero básicamente el azul es microbioma y el rojo es el metaboloma. Aquí en este primer bloque vemos que en el escenario 1 esto nunca se da. siempre se produce un impacto del agente biológico. Lo segundo es que el metaboloma no se ve influenciado tan frecuentemente. Lo tercero que vemos es que los efectos son distintos y si lo comparamos con el tricoderma, con las dos especies de tricoderma, pero distintas cepas, vemos que la respuesta es distinta en los tomates. O sea que a pesar de que son de la misma especie, podemos esperar diferencias. Y por último, pero no menos importante, lo que está en negrita, vemos que hay distintas respuestas en distintas plantas. Así que el contexto depende. Depende de dónde lo introduces. Luego hicimos un último experimento porque nos interesaba el trabajo del mecanismo de utilizar el tricoderma como hongo y trabajamos con el recubrimiento de las semillas de tomate y comparamos los resultados con tratamientos que se habían realizado antes. Es decir, se había aplicado el producto directamente al suelo. Y a la izquierda vemos las semillas tratadas y a la derecha las semillas que no se trataron. Y vemos que el efecto es más o menos el mismo que el que hemos visto en la lechuga. Es decir, que el P9 tiene ciertos efectos. Y aquí vemos las diferencias con el tratamiento con Fusarium, mientras que en los resultados de la derecha vemos que los efectos no están tan claros. Y la flecha roja quiere decir que el efecto fue inferior. Con el impacto de Fusarium no fue muy fuerte en comparación con el caso de las semillas. Esto quiere decir que las semillas dan la oportunidad a que el agente biológico acceda rápidamente, mientras que en las plantas que están en crecimiento la situación no es igual. Así que podemos proponer una sucesión de acontecimientos. El P9 está colonizando las plantas, los tomates y las lechugas. Reduce el metaboloma que aumenta la resistencia de la planta y esto lleva a una respuesta del microbioma. Esto es una hipótesis, no está totalmente comprobada, pero consideramos que esto ocurre así por la reducción de las tasas de crecimiento que vemos a la derecha, que lleva a un aumento de la resistencia a Fusarium. Y esto confirma lo que hemos visto antes en el artículo de 2009 en cuanto al experimento en las patatas. Así que vamos a llegar a las conclusiones. En primer lugar, todos los agentes biológicos, en todos ellos cambió el escenario. El P9 estimula la resistencia en la lechuga y en el tomate, que son especies totalmente distintas, y esto afecta a la planta dependiendo mucho del contexto. En un contexto más amplio puedo decir que también nos gustaría saber cuáles son los efectos secundarios de la inoculación. Siempre inoculamos las plantas, pero no nos damos cuenta de que esto puede tener un impacto en el macrobioma, que es una parte muy importante de las plantas, en el microbioma de las plantas, perdón. ¿Podemos utilizar la microinoculación para controlar el microbioma? Es una pregunta muy importante. Esto sería como un botón de control para controlar el microbioma y esto sería muy beneficioso para la producción. No estamos tan lejos de conseguirlo, pero todavía nos queda mucho por hacer. Tenemos que saber cómo funciona, qué especies influencian y en qué dirección beneficiosa va a ir. Esto quiere decir que hay muchísima gente que colabora en este proyecto, 24 personas en el laboratorio. Les doy las gracias. Y quiero comentar una revista en la que participo. Si están interesados, pueden echar un vistazo. Así que si están interesados en publicar artículos, por favor no duden en comentármelo. Y por último, me gustaría darles las gracias por su atención. Y muchas gracias por haberme invitado a dar esta charla. Gracias.
Muchas gracias por no pasarse el tiempo. The next paper is the biological aspect of the soil health. And we have with us Alberto Acebo from Biome Makers. It's a pleasure for me to be here. I'm going to be where I'm more comfortable, which is here on the ground, near close to the microorganisms. This is where we apply technology of massive sequencing to identify not 1, 2, or 3, but all the microorganisms that live in the soil, bacteria, fungi, to try to understand what they're telling us. That's what Biome Makers, what we do at Biome Makers. That's why I'm not going to present any slides with a petri dish as we've been seeing all day, because microbiome is about this, that all the microbiomes that live in the soil. And the first thing that I have to say is to present an innovation. This is the first time you're going to be seeing it, and the effect that that it's going to have on all of you designing prebiotics, probiotics, all agronomic engineers that are here, how all of you are having an impact on one of the aspects of agricultural sustainability, microbial diversity. You can scan the QR code, and if you do it, you can find a real plot with a big crop rate, A level. And we can see how they relate among each other, how they are connected in a less specialized way. Humans are applying products that are more environmentally friendly, and this translates in this level, in this quantification of biodiversity level, A level. So if I were a farmer today, or an agricultural engineer that wants to understand agricultural sustainability, what can I do? What kind of product and management do I use? Trichoderma from this company, bacilli from this other company. What is the difference, and what is the best one for me? Because the tests, some of them were carried out in Almeria, Andalucía, United States. Which one should I buy, and which one is going to be effective for me? And also that is sustainable and good for the health of my soil. These are the main questions that the industry has to ask itself when applying innovations that we bring here to this kind of event. For this reason, we want to apply a methodology based on bee crop trials. This methodology has a specific protocol to carry out trials, site or field trials, using the microbiome as a reliable biomarker of the effect or management that we use on the field in relation with those microorganisms living there. But to be honest, if I told you all the names of the microorganisms appearing there, and in Biome Makers, we have 15 million of microorganisms that are different and have been identified in different countries, 100,000 samples of soil microbiome collected from all the whole world. You would just find it very difficult. We use advanced technology, algorithmia, to draw information from the microbiome and to allocate it to 4 different indicators: a sustainability indicator, biodiversity of the soil that is linked to the carbon sequestration, because these are the microorganisms involved with the cycle of carbon, and efficiency factors to see the nutrients that are going to the plant, the potential of production of phytohormones vis-a-vis abiotic stress, and the applications that we're generating, how they impact on the risk of disease risk. These trials, these field trials, have to be standardized. We've seen how some companies were presenting some field trials in a different location. Well, literature already shows us how to standardize that process. It's also very important to choose where we're going to apply the product. This is a key question, and how to develop the sampling. We always propose to have a control area and a treated area, to gather samples, to have a baseline before and then after the application to the product, and afterwards to have different timelines and collect replica of the samples. We have 3 different locations, and we can give you information as to when I apply this product, the indicator goes up or down, what is the amount, or qualitative, how much does it go up or down, and whether there is a statistic significance, whether the indicator goes up or down. Do I have to believe in it or not? And it's interesting that after many trials, we always observe the same thing. Depending on the location where the same product is applied, we're going to have different effects. And this is very important. We need to know this. We also, when choosing this location, it's not enough with go to the A experimental plot or B experimental plot or wherever I can. This is an example of an area that Biome Makers is carrying out, because internally we also test commercial products. This is a region in the United States, and what we try to do is to place 10 different trials of products associated to the production of maize or corn in places where they can be made different depending on the environmental and biological conditions. This is a representation of what these conditions are. Temperature of the soil and pH are critical factors. Then we also have organic matter and other indicators that are going to help us locate where these trials are going to be more representative. Today, we have worked with over 70 companies, many of you are here, in over 370 field trials with different products all over the world. This generates a lot of information that can be used. You can use it for different things to scientifically prove the effect of this product on the field in different places, in general terms and in specific terms. Here we have different publications, different papers or journals where we have collaborated with Bayer, BioIberica, and with universities. And here there is a scientific variation of what is happening and how this product has an impact in the life of the soil. We also show an image I'm very proud of, because many of our clients use our studies to project in conferences like this one the beneficial impact of their products, not in the petri dish, not analyzing the genome or in the lab, but on the field, in field trials. You have Bayer, Syngenta, OPL, Alda Energy. We are included in their presentations, in their leaflets, and we are an independent entity that provides this information. They're also used for patents and for registration of products. But What else can be done with this? Well, we've heard a lot about AI, and in biome makers with more than 100,000 samples in our database and with information generated in all the trials on microbiome, we are going to use AI. What for? If you test in different fields and we know the microbiological composition of these fields and how the product has, what impact the product has with that soil biology, and we also know the environmental conditions and the crop where the trial has been conducted, what we can do is get that information from the trials, the information that biome makers has of hundreds of thousands of samples in the databases in the whole world, the information available in public databases from NDVIs regarding climate, types of soils, and put everything in a model based on AI that is generating the projection of what the product of how that product could impact in a beneficial or negative or non-beneficial way in different places in all over the world without conducting trials in those specific places. This is an example of a product that was designed against botrytis with a fungical base. What we saw in the trials is that this product in 4 or 5 locations had been effective in 60% of the places when reducing that risk. If we project that number using AI around the world, we see that in 67% of the places where biome makers has references of microbiome, that product would be recommended against botrytis. In addition of the effectiveness, we can also compare it with the effectiveness of a group of products of which we know what the results of several trials with the same composition. So we see, for instance, that regarding the average of the group, this product is quite much better or a bit better because the average of that kind of groups would be to recommend them in 30% of the places, but this one is recommended in 67% of the places. In addition, since it's a fungicide product, it can reduce the risk of other illnesses or diseases. And in this case, since it's a very specific product for other diseases, it would be less recommended than other products that have a wider spectrum. This is regarding effectiveness at global level, but we can see if it's a fungicide product, how it affects this indicator of sustainability that I mentioned, because if it kills botrytis, it can kill other fungi. In 47% of the cases, this product reduces fungic biodiversity, but it's below the average of the products that we have with reference products of this type. This we can do at global level, but also we can reduce the scale and do it at national level. In what country, in what market would this product be more recommended? We see that in the United States, this product would be recommended in 73% of the places where we have information. As for Spain, only 55% of the times of the places would be recommended. Why? Because in Spain we have different humidity conditions that are different than in the US. So maybe that's why it's less recommended here or more recommended in the United States. These are national level. And what happens if we go to, we analyze the region level? We could also work at region level. And I'd like to finish my talk with this image. This is my daughter, because both biome makers and all of you here that are committed to different agriculture, we're committing to this for a different reason, for new generations to be able to live from agriculture, to be able to take foodstuffs that are produced in a more sustainable way, because improving the health of soils, we're going to generate better products and we're going to improve the health of the planet and the health of future generations. Thank you very much.
40 días por la siguiente. Okay, we're moving to Pilar Truchado. She comes from CSIC. and she's going to talk about composition and modulation of bacterium and resistome of fruits and vegetables, agricultural practices, climate factors, and post-harvest conditions. Hi, good evening. Thank you for coming, and thank you to the organizers for inviting me to this microbiome 2023. I work at CSIC in fruit and vegetables quality, and I'm going to show you the different research lines related to microbiome that we do in my group, both related to bacterium and resistome. We all know microbiome. It's a group of microorganisms in a habitat, but also the enzymes, the metabolites, and the genes, and everything they produce to interact with the environment they are in. In the case of my research, they are focused on bacteria. That's why I chose bacterium. And the habitat I work on is leaf vegetables. In order to determine the composition of this bacterium in this food, we can use techniques dependent on the crop or culturomics, but we have to take into account that just 10% of bacteria are able to grow in a dish. So it is necessary. That's why it is necessary massive sequencing techniques that enable us to know phylogeny and the composition of this bacterium using 16S or metagenomics, directed or non-directed metagenomics, where we can assess the genes and the metabolic routes. And this composition of microbiota can be analyzed through the pre-harvest and post-harvest techniques. There are different factors influencing the plant bacterioma, such as environmental factors and agricultural practices. And within the plant factors, it can be like the type of leaf, etc. We have focused on environmental factors such as solar radiation, and we were able to determine how these pigmented baby lettuce, 3 weeks before harvest, it was covered with a cultivation tunnel, and we saw the changes happening in the bacterium. And the conclusion was that solar radiation was able to modify the variety of this community because change was perceived in the abundance of gamma proteobacteria while increased the relative abundance of alpha proteobacteria. Another research we carried out was related to irrigation. In 2017, the EFSA, the European Commission, based on the opinions of EFSA, defined the 5 main risk factors in primary production, and one of them is water. In order to be able to keep that microbiological quality of water, different strategies can be used, and one of them is the application of disinfection treatments. Up to this moment, most studies have focused if this irrigation water sanitation affect resident bacteria, but the rest of the community of bacteria is never analyzed. If it can analyze the quality of the product, and the research carried out was in baby spinach, in open field cultivation of baby spinach, one of them irrigated with regular water and another one with treated water. First of all, we assessed that the disinfection treatment inactivated the presence of E. coli, and it had an effect on the quality of water. Through 16S, we were able to assess how this affected the composition of this bacteriome, and we were able to see the changes that took place in the structure of the bacteria community, where no changes were seen in water treated with chlorine dioxide, but the composition of the plant microbiome was influenced by the soil microbiota. When we analyzed changes and regarding at phylum level, we didn't find any changes. However, we saw that regarding families, there were changes in some groups, especially those related to Pseudomonaceae and Enterobacteriaceae. This is important because these bacteria are located in some of these plants. In post-harvest, at the moment, we're seeing how different control strategies modulate the composition of microbiota. This is the case of a pathogen that causes very important outbreaks, and that's why we need to find control strategies to reduce this pathogen in vegetable foods. And one of the strategies is the use of bacteriophages. They have antimicrobial properties, and through the Center for Produce Safety, this is from the US, we're going to determine the effect that this bacteriophage has in commercial conditions, together with a fourth range company from the region of Murcia, and we're going to assess changes that happen in the microbiota in two scenarios: a favorable scenario and an unfavorable scenario with abusive storage conditions. The main aim, apart from determining the efficacy of this treatment, is, on the one hand, to see how this microbiota is modulated and if that modulation improves the quality of these vegetables, and to understand the interaction between the bacterial composition and other aspects. Another research line that we are working on at the moment since 2020 is the resistome. This is the group of genes, resistant genes, antibiotic resistance genes that are part of the bacteriome. And at the moment, we are featuring these resistance genes in the agro-food sector, because we do not have data enough to determine these resistance aspects in water, in irrigation water, for example. The WHO has said that bacteria resistance to antibiotics are a world problem, and they're going to cause lots of deaths in the future, more than cancer. Here we see the water treatment plants, and this is the place where we find high concentrations of antibiotics, and they favor the transfer from the bacteria to genes. Due to the lack of water in Mediterranean countries, here regenerated water is widely used, and despite the fact there's a legislation, reclaimed water, there's a lack of knowledge on the presence of these bacteria and these genes resistant to antibiotics in this type of water. In 2021, the EFSA, in a scientific opinion, assessed the different transmission paths of genes with resistance to antibiotics in the production of food. And here we find irrigation water. According to this idea, and thanks to funding of a project called Red Water, we want to make an assessment of efficient water disinfection technologies compatible with sustainable water reuse approaches in circular economy agri-food production systems. This is a project that's coordinated with the University of León, and within the project that the CEBAS is part of, the main aim is going to be what the genes are going to be, what types they are, the concentrations they can find on any the current treatments that are applied, the water treatment plants can eliminate or reduce the presence of these agents with resistance to antibiotics, so that we can assess if the use of this reclaimed water can spread genes and bacteria to the environment, and in case they were accumulated in the soil, if they can constitute a risk for consumers' health. This project started in December 2022, and the first results is that the efficacy of water disinfection technologies for reclamation of irrigation water have been determined for tertiary treatments, such as UV treatments, and we see that there's a reduction of these resistance genes at around 3 to 4 logarithms. Although resistance in the water values are kind of high. So we have to see what happens with the plant and with the soil. And to sum up, these research lines I'm working on have focused on researching the bacteriome and the microbiome to offer food that is safe, sustainable, and quality for consumers, because they're going to determine and they're going to help us to improve the quality of these vegetable foods and to guarantee their safety. Finally, I would like to introduce my research group from CEBAS, the Microbiology and Quality of Fruit and Vegetables group, and I want to thank them for participating in this research. Thank you very much for your attention.
Thank you, Pilar, for your presentation. It was very interesting. Now we're going to welcome Ignacio Villalba from MAFA, and he's going to talk about the application of gram-negative bacteria for the optimization of the growth of horticulture cultivation. Have we got Ignacio with us here? Welcome. Good evening. Can I start? Okay. Good evening. I'm Ignacio Villalba, a researcher, microbiological researcher from MAFA. I'm going to talk about the application of gram-negative bacteria for the optimization of horticulture cultivation. MAFA is a company that is part of the Sierra Sur group, one of the main top 10 companies in Andalusia with 150 employees. In MAFA, we produce and formulate 20 tons per day. It has its own microbiological lab and has a capacity of fermenting 300 liters. And we collaborate with public institutes as CSIC and ICA in microremediation of fungi. We're going to talk about soil, this living element, but everything cultivated has they have something in common, rhizosphere. This area is highlighted because it's very much alive. 5% of the material is made of microorganisms, and here there is some symbiosis between the plant and microbiota, especially in the root, being this one of the most diverse ecosystems that we have. However, well, the thing is that today, with the loss of cultivated areas and the lack of resources as water and degradation of water, it is a great problem to be able to replace or to supply the market with everything we need. So from MAFA, we commit to a PGPR, plant growth promotion of rhizobacteria. The Pseudomonas chlorophyllus CST9 is a gram-negative bacteria that is mobile and has lots of benefits with APS, expopolysaccharides and auxins. It goes against fungal phytopathogens, producing therefore of iron. Iron is key, and it's key for enzymes like in the cell breathing that gives energy to the plant and the cell. Siderophores are have nitrogen and oxygen. In fact, siderophores produce pyoverdine, and we see it there with its own structure. When pyoverdine is created, this directly inhibits some phytopathogens because it is the disruptor of membranes. It can also meet with the iron to create a more soluble compound. This favors the absorption and the stimulus of the plant that has better nutrition, but apart from this, enables the better growth of local microbiota. What does this mean? That the soil, the root, is more colonized, so it's more complicated for phytopathogens to colonize the environment, avoiding infections. Apart from all this, it favors the benefit of local microbiota in that plant. In addition, it can solubilize phosphorus, a key element, because it's in the DNA and is part of one of the most essential molecules, ATP, like an energetic currency. The way that this can be solubilized can be direct with phosphatase or indirectly. It produces organic acids, and this acidifies the soil, and this solubilizes this element that is phosphorus in the shape or in the form of phosphate. I'm going to give an example now how we observe this pyoverdine provides color that is like green, fluorescent green. Solubilization of phosphorus produces halos and generates biofilm. You can see the different layers. To talk about biofilm, I have to talk of expopolysaccharides, EPS, with amines and phosphates, and they form an extracell matrix, and this enables the correct creation of biofilm, a protective barrier that is physical around the root, enabling the solubilization of phosphorus and concentration of iron, among other. And this in turn enables the production of sensing quorum. This is very important because this is the communication produced between microorganisms, among microorganisms and microorganism and environment. This coordinates a microbial growth and a metabolic function that is very important to prevent diseases. Here there is a clear example where a phytopathogen called Fusarium meets the plant, and if there is no quorum sensing with AHL, they will only receive an antifungal response, those bacteria in contact with the phytopathogen, slow response and with little concentration or less concentration. And this will develop pathogens. But if the bacteria relate among themselves, they would know that there's a phytopathogen and there will be a systemic response, coordinated, quick, and diverse. So this would avoid the installation of the pathogen. In addition to this, we studied how our product affects the soil. We can see three graphs, A, B, and C. A and B have a common structure with no treated and treated after 20 days and after 90 days. In the graph, we studied the presence of pseudomonas. There's a bigger presence of pseudomonas. So 20 days after, they are still present and they have an effect. In the B graph, there was a test to see genetic diversity, to see how it increases. In the C graph, there is a break cultist. We study this diversity and we can see that there are two different groups that do not interrelate. So these pseudomonas have an impact on the rhizosphere. So at the beginning, on the right, you can see that there is higher diversity that is reduced later. It can sound counterproductive, but no, because microbiota is optimized, and I'm going to explain why. To this end, we created a study, a metagenomic microbiota test. We see the difference between treated and non-treated. The darker is the higher presence of microbiome. We can see that PGPR are there, and in the non-treated, we have Aeromones, Varicella, Clostridium. These are human pathogens. So this is why it optimizes microbiota. Apart from all this, pseudomonas produce auxins through the tryptophan pathway and the acetylation of the latter. It's also capable to induce the expression of auxins, endogen auxins of the plant. This qualitatively increases the amount of auxins in the plant. So this enables to improve the epigenetics of the plant, producing substance like antioxidants, and this reduces to a larger resistance of the plant to generate diseases and more tolerance to water, salt, stress, and high temperatures. Apart from the typical function, which is, as you can see, the development of the root. You can see it with ST9 and non-treated. And here we see more biomass, more relation of the root with the vis-a-vis the control sample. Apart from this, we wanted to see how our pseudomonas are capable of affecting the plant. So we carried out a trial on the transcriptomics of the plant. How much can a gene become more more expressed? And what we did was we grouped them in two groups. The first one is in blue. These are genes that are related to ethylene. And the overexpression of these genes increases the tolerance to water stress and salt stress. But then there's another kind of group in yellow. This group of genes is related to the signaling and regulation of auxins, and these proves what I said before, that pseudomonas induce auxins in plants. As a practical example, these two plants were treated with salt and water stress, and we can see the difference in physiological terms of the plant with treated with ST9, and this is owing to what I just explained. In addition to all this, pyoverdine, the production of protease, and all this, these pseudomonas are capable of having an antifungic and it can produce substances that degrade this phytopathogenic fungi like HPR and PCA. This is in a petri dish. We carried out a trial, and we see that pseudomon does not touch the fungus. There is like a protector barrier, and the fungus runs away and colonizes on the opposite direction. And this means that it has an effect on the real control, and it runs away from it. It cannot be colonized. We tried with the different elements that you can see on the screen. We also tried it with botrytis cinerea, phytium, and penicillium. Phytopathogens only on the petri dish first. Then we can observe after 6 or 8 days the petri dish treated with pathogen and our product, observing the same result. This result was so good that we said, let's escalate it and do something more real as to what happens on the field. And to this end, what we used was real tissue of pepper. This is the water treated with water. In the image in the middle, we see the infection of the phytopathogen because it was inoculated, and at the bottom one, we saw that this same infection by phytopathogen, but treated with ST9. So what we can see is a clear difference between the leaves, because the ones in the middle have like a wound and the other ones don't, and this is because the fungus cannot affect the whole leaf owing to the antioxidant, the pyoverdine, chemical compounds. In fact, having said this, I would like to say that the soil is a living element that is very important that we should take care of. The use of the latter is a great challenge. PGPRs are the solution to these current challenges, and from MAFA we propose ST9, as I showed, ST9 as a solution to this problem. Thank you very much, and if you have any questions, I'll be happy to answer.
Thank you very much, Ignacio. We're moving to the last presentation. It's by Rosa Domenech from ITENE on how to get nutritious food for all, microbiome and agriculture. Welcome, Rosa. Buenas tardes a todos. Me gustaría dar las gracias a los organizadores por invitarme a venir al Congreso como ponente. Me llamo Rosa Domenech y trabajo en el Centro de Investigación de ITENE. Soy directora del Departamento de Microbiología. Vengo a presentar el proyecto de Horizon Europe que se llama Tribiome, que Para no desarrollar el nombre del proyecto, que es muy largo, lo he denominado con el nombre del proyecto, que se llama Trivium. Este es el contenido que voy a tratar. Voy a presentaros el consorcio que participa en el trabajo del proyecto. Luego presentaré las necesidades del proyecto, las necesidades de la sociedad y el objetivo del proyecto. Os mostraré las acciones que se van a tomar para conseguir los hitos del proyecto, las innovaciones que se van a producir durante el proyecto y los resultados que se esperan al final. Los objetivos se van a conseguir utilizando la experiencia de cada uno de los socios del consorcio. El consorcio consta de 6 centros de investigación. Hay 3 centros de investigación, CSIC e ITN en España y Luque de Finlandia. Hay 3 universidades, la Universidad de Bolonia en Italia, la Universidad de Pretoria en Sudáfrica y la Universidad de Burgos. Esta última es la líder del hub digital de ganadería, medio ambiente, agricultura y silvicultura. Hay 2 organizaciones no gubernamentales, una de Italia y Asaja de España. Las 2 representan a las asociaciones de agricultores y los socios de innovación europea. Hay 4 pymes: Val Genetics de España, Simavi de Rumanía, Partícula de Croacia y Bagralim de Bélgica. Esta última representa a los socios innovadores de la industria de la alimentación. Y por último hay una gran empresa que se llama Fertiberia, que es líder en fertilizantes. Una vez que he presentado el equipo de trabajo, nos vamos a centrar en el problema que queremos resolver a través del proyecto. En los últimos 2 siglos, la población mundial ha aumentado de 1.000 a 10.000 millones debido a los avances médicos y la mejora de la productividad agrícola. En un futuro breve se espera que la población alcance, como he dicho, pase de los 8.000 a los 10.000 millones de habitantes en 2050, según los datos más recientes de las Naciones Unidas. Esta cifra puede que dé lugar a problemas de recursos y de contaminación. Y por ello, hace varios años surgieron distintas estrategias. La estrategia de la granja a la mesa, que evita el uso de pesticidas químicos para evitar las pérdidas de nutrientes, y trabaja con el uso de antibióticos en los animales en la ganadería. El objetivo es reducir el último objetivo de reducir las emisiones netas de gases de efecto invernadero a 0 en 2050, mientras se garantiza la prosperidad de la economía europea. Esto forma parte de la Agenda 2030, que se lanzó en 2016. Esta política ofrece un marco para transformar los sistemas de alimentación en sistemas resistentes para garantizar que todos tengamos acceso a alimentos sanos y asequibles. El suelo tiene un papel importantísimo para la agricultura y sabemos que el microbioma tiene un papel fundamental para mantener la vida en la Tierra, en la fijación de nitrógeno o del metano y la fertilización y reducir los gases como el metano. El microbioma está muy relacionado con la salud humana. Esto tiene un papel muy importante en la salud humana y vegetal. Así que es más importante, cada vez es más importante entender la relación entre los microbiomas, ya que tienen la capacidad de mejorar la situación y la circularidad. El objetivo del proyecto es profundizar el conocimiento del microbioma del suelo y de las plantas, estudiando la variabilidad según distintos estreses bióticos y abióticos, con el objetivo de llevar a cabo un modelo que nos permita seguir investigando nuevas herramientas para modular y mejorar la producción agrícola, así como para mejorar la producción agrícola para obtener alimentos nutricionalmente avanzados, mejorados y más nutritivos, con un impacto positivo en el microbioma actual y en los humanos y en los animales. El objetivo del proyecto tiene una perspectiva, desde distintas perspectivas. Aquí los socios trabajarán con el Foro Internacional de Bioeconomía para conseguir los objetivos globales. El trabajo técnico consiste en modelar microbiomas para aumentar la dinámica y mejorar la calidad y la productividad. Y después aislar los moduladores con efectos relativos, garantizando la circularidad a través de procesos de valorización. Por último, las nuevas herramientas desarrolladas se validarán en humanos y en animales. Es interesante destacar que las tecnologías que se desarrollan en este proyecto vienen de la investigación básica de la fase TRL3 hasta la TRL6 . Así que las principales innovaciones que tendrán lugar en este proyecto están alineadas con el marco 2030. El trabajo de tribioma aumentará la calidad nutritiva de los alimentos y favorecerá la digestibilidad de los humanos y reducirá la malnutrición y los problemas de hambre. Aumentará la resistencia al estrés biótico y abiótico. Valorizará los residuos agrícolas para producir preparaciones ricas en moduladores, promocionará una economía circular. Y la solución que propone Tribioma se validará y demostrará en 3 regiones, que son España, Italia y Sudáfrica. Así que los resultados que se esperan para cada uno de los participantes en la cadena de valor de los alimentos son en el sector de la agricultura: diseñar, integrar e implementar estrategias de manipulación y gestión del microbioma. Para el aspecto académico y los investigadores, obtener una mayor comprensión de la dinámica de las plantas y del suelo. Para los consumidores, producir soluciones más sostenibles y económicamente favorables para mejorar las prácticas agrícolas. Y en cuanto a las autoridades públicas, Tribioma desarrollará una plataforma para incorporar las comunidades que se encuentren de microorganismos en el proyecto. También se espera una mejor cooperación y comunicación a través de todos los actores, todos los participantes de la cadena alimentaria y la red de toma de decisiones. Espero que os haya convencido de la excelencia y la innovación que vamos a aportar y a contribuir a la sociedad del microbioma. Gracias por su atención.
Muchas gracias, Rosa, por este proyecto tan interesante. Okay, as Bugs Bunny would say, that's all, folks. And it's all regarding papers, because I have to remind you that now we can have a gin tonic and continue our networking in the hallway. Tomorrow we'll continue, and I don't know whether it'll be better, but at least it'll be as good as today. Thank you so much. See you tomorrow.